Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod.
Good morning and welcome everyone to the last day of the Regulatory Education for Industry Annual Conference 2022. This conference is conducted in collaboration with the FDA's Small Business Industry Assistance Program as an educational event on various regulatory topics in medical product development. I would like to also welcome everyone to the second day of CBER Track. Today you will hear presentations from the Center for Biologics Evaluation and Research. But before we delve into today's topics, let me once again take care of the administrative and continuing education information and disclosures. If you have participated with us since Monday, you've heard this multiple times, but please bear with me another few minutes. I have to say all of this information again. As was already mentioned, this conference offers up to 32.5 interprofessional continuing education credits, or CE credits. The obtained certificate of attendance may be used in support of credits for the Regulatory Affairs Professional Society, the Society of Clinical Research Associates, the Association of Clinical Research Professionals, and the Society of Quality Assurance, as well as continuing education credits for physicians, nurses, and pharmacists. In order to claim your CE credits, all participants must attest to their attendance and complete faculty evaluations and the final activity evaluation via the CE portal, and that is ceportal.fda.gov. For multi-day activities, participants must attest to their attendance and complete the faculty evaluation each day. Final activity evaluations must be completed within 2 weeks after the activity, and the deadline for this conference for evaluation completion is June 24th. But of course, besides the need to demonstrate attendance, your evaluations are very important for our planning of future educational conferences, and we greatly appreciate your feedback, comments, and suggestions. At this point, I would like to remind pharmacists and pharmacy technicians, and I'm sure you've heard this before, but I have to repeat it again, please provide your correct NABP profile and date of birth information in the required format, which should be the month and then the day, to ensure that you receive credit for this activity. Pharmacists are recommended to log into the CPE monitor 8 weeks after the last session of the activity to ensure that your credit is captured correctly. Each track of the conference will have its own survey evaluation link. Each night, SBIA program has been sending, and tonight they will send again, an email to all of the attendees who logged into today to listen to our sessions. The email will have information on the CE claim codes and will contain the survey and evaluation links. Again, as a reminder, the CE credits are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend this conference via Adobe. And now to the disclosures. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single company. Continuing education faculty speakers are required to disclose when products or procedures being discussed are off-label, meaning they're not FDA approved, and if there are any limitations on the information that's being presented. Finally, all CBER speakers who are presenting today have nothing to disclose. And now we can start our first session of the day, in which we will discuss CBER's regulatory approach to safety surveillance, signal detection, risk management, and assurance of safe use of biological products in the post-approval period. The first CAR-T product was approved in 2017, and with it, a new risk evaluation and mitigation strategy was put in place and became the set of risk mitigation measures that have been adopted to ensure safe use of the subsequently approved CAR-T products that are currently available as treatments for various hematological malignancies. So our first presenter of the day is Dr. Meghna Alimchandani, who serves as the Deputy Director of the Division of Pharmacovigilance in the Office of Biostatistics and Pharmacovigilance in CBER. In her work, she focuses on pharmacovigilance planning, passive and active post-marketing safety surveillance for products regulated by the Center, formulation of REMS programs, and oversight of the pharmacovigilance reviews for biologics license applications. Dr. Alimchandani will give us an overview of the pharmacovigilance activities and will describe examples of post-marketing studies and risk evaluation and mitigation strategies for CBER-regulated products. Our second presenter in this session is Dr. Joyce Abidi, who currently serves as the health scientist in the CBER surveillance team in the Office of Biostatistics and Pharmacovigilance. Dr. Abidi holds a PhD degree in cellular and molecular medicine. Her postdoctoral research was focused on optimizing new therapeutic strategies and personalized medicine, aiding in the stratification of patient subgroups, particularly patients with oncological conditions. Here at FDA, Dr. Abidi works on building and utilizing a post-marketing surveillance system for monitoring the safety of biologics regulated by CBER, specifically focusing on how to harness electronic health records to complement FDA's post-marketing surveillance activities in building the foundation of a national biologics surveillance system. Dr. Abidi will tell you all about it during her presentation, as she will be describing the biologics effectiveness and safety system in CBER. Again, while listening to the presentations, please send us your questions through the chat box. We will have the Q&A portion right after both presentations. Dr. Alimchandani will start us off.
This is Meghna Alimchandani from the Center for Biologics Evaluation and Research. And today I will be providing an overview of post-marketing safety surveillance in CBER. We will touch on the following topics during this talk: Pharmacovigilance Plan or PVP, Routine Pharmacovigilance, Safety-related post-market studies, Risk evaluation and mitigation strategies, Signal detection and evaluation. Our first topic is the Pharmacovigilance Plan or PVP. This slide provides an overview of the key decisions in pharmacovigilance planning. Pharmacovigilance is defined as the science and activities related to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problems. The PVP contains the sponsor's assessment of safety specifications for their product based on the clinical safety database. The manufacturer lists important identified risks, important potential risks, and important missing information. This safety specification is intended to help industry and regulators identify any need for specific data collection and also to facilitate the construction of the pharmacovigilance plan. Our job at FDA is to review the safety data and the proposed pharmacovigilance actions and make a decision about the adequacy of the PVP. The items listed on the right side describe various components of a PVP. Now, all pharmacovigilance plans include routine pharmacovigilance. Routine PV is mandated by the regulations for all licensed products, and we will discuss routine PV in the next slide. The next two pharmacovigilance activities may be required in addition to routine PV, depending on the safety profile for the product. The sponsor may conduct post-market studies, as post-marketing requirement studies or post-marketing commitment studies to further assess serious risks. FDA may require mitigation under a REMS program to ensure that the benefits of a product outweigh the risks. At the bottom of the slide are listed the FDA guidance documents for reference on this topic. Now we will discuss routine pharmacovigilance. Through routine pharmacovigilance, FDA performs continuous post-market safety monitoring for all licensed products. This is mandated by the regulations under 21 CFR 600.80. Under passive surveillance, spontaneous adverse event reports are submitted by the public and by manufacturers. There is voluntary adverse event reporting by healthcare providers and the public. There is mandatory adverse event reporting by manufacturers as required under the regulations. There are two types of manufacturer spontaneous adverse event reports. Expedited or 15-day reports have to be submitted for all serious and unlabeled adverse events. Non-expedited reports are submitted for all other adverse events, that is, non-serious events and for serious labeled events. The manufacturer also submits periodic safety reports, which contain aggregate review of safety data from different sources and the sponsor's assessment for any safety signals that were identified in the reporting interval. Periodic safety reports may be submitted in different formats. For example, periodic adverse experience report, periodic safety update report, or the periodic benefit risk assessment report. The FDA Adverse Event Reporting System, or FAERS, is the pharmacovigilance database that contains adverse event reports and medication error reports that were submitted to FDA for therapeutic biological products. The database is designed to support the FDA's post-marketing safety surveillance program for drug and therapeutic biological products. Continuous monitoring of the safety profile of approved products includes signal detection, evaluation, and liaison with regulatory authorities, including global pharmacovigilance and other public health agencies. The next couple of slides will touch on qualitative and quantitative analysis for passive surveillance data. Some of the aspects of qualitative review at the level of the individual case safety report, or ICSR, includes the following: looking for patterns to detect signals of adverse events, looking for unusual clustering or trends in adverse events, biological plausibility, consistency with known effects of the product, absence of alternative explanations, such as concomitant medications or underlying disease, and positive re-challenge reports. A series of cases for the same product or product type, similar in the reported event and fitting a case definition for the event, becomes a case series. Demographic and clinical patterns from case series can be analyzed and reviewed to further evaluate the potential association between the event and the product. This and other best practices are discussed in the FDA Best Practices document, for which the link is provided at the bottom of the slide. Quantitative review includes data mining and screening for disproportional reporting, which is further discussed on the next slide. Reporting rate is the number of reports received over a given time period. Reporting rate can also be expressed by the number of reports over the number of doses of a product distributed, if the distribution data is available. Reporting rates may be compared to the background rates for an adverse event of special interest. Reporting rates are not an incidence. We must keep in mind that passive surveillance is limited by significant underreporting, so actual occurrence of an event may be much higher than the reporting rate. Reporting rate can also be biased by the age of the product, the public interest in the event, the seriousness of the event, and various other factors. Data mining helps us assess the proportion of reports with a specific product adverse event pair compared to the background database of product event pairs. This graph shows adverse events reported for a specific product with high statistical scores, alerting staff that further investigation may be warranted. Very important to keep in mind, an elevated score does not mean there is a causal association between a product and event. Data mining alerts are hypothesis generating and further evaluated. This slide describes some of the strengths and limitations of passive surveillance systems like FAIRS. Passive surveillance systems are described as the nation's early warning system for product safety. Strengths include the ability to rapidly detect potential safety issues, potential detection of rare adverse events, geographic diversity, and capability to monitor lots if lot information is provided in the reports. The right side of the slide lists important limitations of passive surveillance databases. Reports may have missing or inaccurate data. Reported diagnoses are not verified. There is underreporting. There may be reporting bias and stimulated reporting. There is absence of a control group for comparison. Limited information in the report may preclude assessment of a causal association, and passive surveillance is not likely to detect long latency events. These are some examples of safety signals from surveillance activities over the years. On the left is a 2016 921 posting of a potential signal of serious risk identified from FAIRS. This posting, which is known as a 921 posting, was mandated by FDA, Food and Drug Administration Amendment Act of 2007, Title 9, Section 921. Under the regulations, FDA is directed to conduct bi-weekly screening of FAIRS database and post newly identified safety signals on FDA's website. This 921 posting was for a severe product, Imlygic, which contains a genetically modified herpes simplex virus type 1, and the serious risk was related to disseminated herpetic infection. On the right top corner of the slide is a link to a CBER web page that posts information on immunoglobulin voluntary lot withdrawals due to increased reports of allergic reactions associated with certain product lots. On the bottom right corner of the slide is an example of an FDA safety communication and mandated safety label change that occurred back in 2013 to add a boxed warning for thrombosis for the immunoglobulin product class. Now we come to the first challenge question. Routine pharmacovigilance for biological products includes A. Adverse event reporting in accordance with 21 CFR 600.80. B. Submission of expedited 15-day reports for serious and unexpected adverse events. C. Submission of non-expedited reports for serious, unexpected, and non-serious adverse events. D. Submission of periodic safety reports at quarterly intervals for 3 years post-approval and at annual intervals thereafter. E. All of the above. And the correct answer is E. All of the above. Our next topic is safety-related post-market studies. So here we are referring to studies that are being conducted specifically to evaluate safety or further investigate a safety issue that is associated with the product. A safety study must have a primary safety endpoint. Examples of safety-related post-market studies include an observational epidemiology study, a pregnancy registry, a survey, or a study using population-based data sources, such as claims data or electronic healthcare records. Safety studies may be conducted by the sponsor or by the government, FDA or CDC. There are three types of sponsor studies: post-marketing requirement, PMR studies, post-marketing commitment studies, PMCs, which are agreed upon studies between FDA and the sponsor, and voluntary studies. FDA also conducts active surveillance studies. In CBER, we have the Biologics Effectiveness and Safety BEST initiative. My colleague, Joyce Obidi, will be speaking further on CBER's BEST initiative in the next presentation. This slide goes over the regulatory considerations for safety PMRs. Section 5050 of the Federal Food, Drug, and Cosmetic Act was added by FDA, Food and Drug Administration Amendments Act of 2007, FDA, and these regulations give FDA the authority to require certain post-marketing studies and clinical trials for any or all of the following three purposes listed on this slide: to assess a known serious risk, to assess signals of serious risk, or to identify an unexpected serious risk when available data indicate the potential for a serious risk. Before requiring a post-market study, FDA must find that adverse event reporting and the active post-marketing risk identification and analysis system will not be sufficient to further assess the serious risk. Similarly, before requiring a PMR for a clinical trial, FDA must find that a study will not be sufficient to achieve these same purposes. Definitions for clinical trial versus a study are found in the FDA guidance document that is listed at the bottom of the slide. Here are some screenshots of safety PMRs for CBER-regulated products that are excerpted from the approval letters. I wanted to point out that PMRs and PMC studies are described in the approval letters, which are posted on FDA's website and available to the public. The study milestones are also listed in the approval letters, as you can see here. FDA also has a website that is updated regularly with regards to the status of the study. For example, is the PMR ongoing, or has it been completed, or is it delayed? This brings us to our second challenge question. Safety-related post-marketing study may include A, a post-marketing requirement study under FDA, B, an agreed-upon post-marketing commitment study, C, a voluntary sponsor study, D, an active surveillance study using population-based data sources, E, all of the above. And the correct answer is E, all of the above. Our next topic is Risk Evaluation and Mitigation Strategies, or REMS. FDA created Section 5051 of the FDNC Act, which establishes FDA's REMS authority. FDA determines whether a REMS is necessary to ensure that the benefits of a drug outweighs its risks. REMS is a required risk management plan that can include one or more elements. FDA can make the determination about a REMS at the time of initial approval or post-approval when FDA becomes aware of new safety information. FDA may also require elements to assure safe use, or ETASUs, as part of a REMS. ETASUs can include patient monitoring, or certain safe use conditions, or certification of healthcare settings. An ETASU may be required if the product has been shown to be effective but is associated with a specific serious risk and can be approved only if, or would be withdrawn unless, such elements are required as part of a strategy to mitigate a specific serious risk listed in the labeling of the drug. This next slide shows the factors in determining when a REMS is necessary. The regulations require FDA to consider the following 6 factors in making a decision about whether to require a REMS. The FDA guidance document on this topic is listed for reference at the bottom of the slide, and I'm going to pause for a minute so you can read through the different factors that are considered in making the determination whether a REMS is necessary. The next slide provides some examples of CBER REMS programs. CBER has approved REMS programs for the chimeric antigen receptor, CAR, T-cell therapy product class. We currently have REMS programs for Kymriah, Yescarta and Tecartus, Breyanzi, Abecma, and Carvykti. Carvykti is the most recent REMS program that was approved earlier this year. On the left side of the slide, there's a screenshot which is an excerpt of the description of the REMS program from the approval letter for Carvykti. And I'll pause here for a minute for you to skim the information here. Our last topic pertains to signal detection, evaluation, and verification. We will go through a stepwise depiction of signal detection, evaluation, and verification process, which finally may culminate in regulatory action. The top row of boxes shows the various sources of data that we review regularly to detect new safety issues. This is signal detection. Post-market safety issue is defined broadly as information from one or multiple sources which suggests a new potentially causal association or a new aspect of a known association between an intervention and an event or set of related events that is judged to be of sufficient likelihood to justify verificatory action. The sources of data are listed in the top boxes. From pre-licensure, we have clinical trial data. In the post-licensure phase, we have spontaneous adverse event reports in the FAERS database. We may get information from active surveillance using large electronic healthcare databases. We may also receive information from observational studies or clinical trials in the post-licensure period. REMS assessment reports provide an additional source of information. Other sources of information include published literature and foreign regulatory agencies. Once a new signal is detected, it is further evaluated in various ways. This is called signal refinement. Constructing case series from the post-market data or assessing rates of an association between a product and event in active surveillance sources, we further define and establish a potential signal. Finally, if feasible and the signal warrants more definitive assessment, we can conduct formal pharmacoepidemiologic studies using claims-based sources, often with chart review or observational epidemiology studies to further test the hypothesis. All of this culminates in regulatory actions or other risk mitigation or other risk management actions. This can include communication, label changes, recalls, etc. So this slide lists various risk management actions. Revision of the package insert is generally considered one of the primary means of risk management and communicates new information about product risks or adverse effects to physicians and also to patients. Labeling changes may be voluntarily proposed by the sponsor, or FDA may require safety label changes. We spoke earlier about Section 921 postings. There are various ways of public health notification. Manufacturers may voluntarily elect to issue Dear Doctor letters. Other ways of communication could include publication or presentation at professional meetings. FDA can conduct inspections of a facility to gain more complete information about processing or manufacturing at a site, if that might be related to a safety concern. We could have product withdrawal or product recall. And as we mentioned before, if there is new safety information in the post-approval period, then FDA may require a REMS, FDA may require post-market studies, or there may be additional post-marketing commitment studies, active surveillance studies using population-based data sources. And as I mentioned before, communication also happens through professional meeting presentations and peer-reviewed publications. This brings us to our third and last challenge question. FDA has the authority to require safety-related post-marketing studies or clinical trials, PMRs, require sponsors to make safety-related label changes, require sponsors to develop and comply with risk evaluation and mitigation strategies on REMS. True or false? Oh, this is true. All of this is true. This is all falls under FDA's regulatory authority. The next few slides will summarize the information in this presentation. This is a summary of FDA's surveillance process. We started with reviewing the sponsor's pharmacovigilance plan as part of review of an original or supplemental biologics license application. The PVP identifies safety signals in the pre-market phase and informs pharmacovigilance actions for the post-market phase. In addition to routine pharmacovigilance, FDA may require post-marketing studies as PMRs or risk evaluation mitigation strategies. In addition, the sponsor may conduct post-marketing commitment studies or additional voluntary studies. FDA may also undertake active surveillance post-market studies using claims data or electronic healthcare record data to address product safety issues. And as I mentioned before, this topic will be further discussed by my colleague Joyce Obidi in the next presentation. Despite rigorous safety evaluation during pre-market phases of clinical development, post-marketing safety monitoring is necessary due to the limitations of clinical trials. In summary, FDA is conducting continuous safety monitoring for all licensed products. Post-marketing surveillance includes many approaches, including passive and active surveillance. FDA may require post-marketing studies by sponsors. FDA may require safety label changes. FDA may also require risk evaluation and mitigation strategies or REMS. And finally, new databases have expanded population-based surveillance capabilities that will be described further in the next presentation by Joyce. Our goal is to ensure safe and effective products. That concludes my presentation. Thank you for your attention, and I will respond to questions following the next presentation.
Hi, today I will share an overview of the CBER Surveillance Program, the Biologic Effectiveness and Safety Initiative, BEST, and how it is used in regulatory work at CBER. By the end of this presentation, you should be able to explain the purpose and scope of the CBER Surveillance Program and BEST, distinguish the unique characteristics of CBER products and how they influence approaches to post-market surveillance, and describe BEST capabilities that are available for use by CBER product review offices. This presentation is divided into 4 segments: real-world evidence and real-world data. I will then go into a high-level overview of the CBER Surveillance Program. I will then discuss the infrastructure of the BEST initiative. And lastly, I will then show you how we use this for regulatory work at FDA. Let's start with some definitions. Real-world data, RWD, are data relating to patient health status and/or the delivery of health care routinely collected from a variety of sources, such as electronic health records, claims and billing data, data from product and disease registries, and patient-generated data, including in-home use settings. Real-world evidence is the clinical evidence regarding the usage and potential benefits or risks of medical product derived from analysis of real-world data. Real-world evidence can be generated from different study designs, including but not limited to randomized trials such as large simple trials, pragmatic trials, and observational studies. How does this all fit into our regulatory work here at FDA? The overall scope of a real-world evidence program is to evaluate the potential use of real-world data to support changes to labeling about product effectiveness and safety, including adding or modifying an indication, adding a new population, or adding safety information. Some considerations in evaluating real-world data and real-world evidence is whether the real-world data are fit for use, provide adequate scientific evidence, and meet FDA's regulatory requirements. So let's delve a little deeper in what we mean by fit for use, since this is a phrase you may hear more frequently when discussing real-world data and real-world evidence. So we'll start with relevance. The overall assessment of relevance should determine whether the existing real-world data source is adequate for evaluating the performance of a device or product in the identified regulatory context. Since data are used for non-regulatory purposes, FDA will assess whether the source is sufficient to be used for regulatory purpose. This includes assessing the accuracy of the data, the completeness of the data adequacy to address the regulatory question, and the need for review or adjudication of specific outcomes of interest. Then we have reliability, how data were collected and that errors were minimized through data collection and analysis. So we have data accrual. Was there a common data capture form? Data assurance. Are there established data quality control processes? Here are several ways that real-world evidence have been used in regulatory decision-making here at FDA. One example was assessing the geographic incidence of babesiosis to aid in developing a blood donation guidance. Another example, it was estimating the rate of transfusion in high-risk populations for Zika risk assessment in vulnerable populations such as pregnant women, immunocompromised, and the elderly. Here are some other examples of how real-world data can be used to support regulatory decisions. One in particular is it being used to complement adverse event surveillance. For example, it can be used to conduct studies to refine and evaluate signals generated in the post-market period, such as by VAERS. Time for a poll question. The scope of a real-world evidence program is to evaluate the potential use of real-world evidence to support changes to labeling about drug product effectiveness includes A, adding or modifying an indication, such as a change in dose, dose regimen, or route of administration. B, adding a new population. C, adding comparative effectiveness or safety information. Or D, all of the above. The answer is D, all of the above. All of these are part of the scope of a real-world evidence program, which is using real-world evidence to support changes to labeling, and that can be adding a new population, adding or modifying an indication, or adding safety information. Now we're going to move on to the CBER surveillance program, and I'm going to give you a little overview. The mission of CBER is to ensure that biologic products approved by FDA are safe and efficacious. One of CBER's plans for fulfilling this mission is through the CBER surveillance program. The overall vision of this program is to create and utilize an active surveillance system for monitoring the safety and effectiveness of these products, which include vaccines, human tissues and cellular products, gene therapies, blood components, and blood-derived products. Through multiple contracts and various partnerships, which make up the CBER surveillance program, CBER works with a diverse group of epidemiologists, data scientists, clinical experts to conduct active surveillance studies. There are three components of the CBER surveillance program, which include federal partners, such as CMS and VA, academic partners, and then the Biologics Effectiveness and Safety Initiative, BEST, which includes administrative claims data and electronic health records data. Today, I will tell you a little bit more about the BEST initiative. One of the biggest questions we receive about BEST is, why was BEST created? When it comes to the surveillance of biologics, biologics have unique characteristics, and as a result, their surveillance requires specific capabilities that I will expand on in the next 2 slides. Some unique characteristics of biologics include the fact that vaccines are administered to healthy populations in children, which means there's a very low threshold for the risk of adverse events. In addition, there's the continuous monitoring of the blood supply and the occurrence of emerging infectious diseases. This particularly requires access to EHR data and, in some cases, blood bank. All right, now we're going to talk a little bit more about the infrastructure of the BEST initiative. In order to expand and enhance the BEST initiative and to customize its components for the surveillance of biologics, the following components are required. An EHR network, which provides access to granular clinical data that's not available in administrative claims data sources. A reduced data line for about weeks, not months, which allows for monitoring of newly approved products. On-demand analytic capabilities, where more complex studies can be conducted without the use of prepackaged tools and programs, access to large claims databases, an expandable common data model that can adapt to our surveillance needs very quickly, and lastly, easier, faster, and affordable access to medical charts for validating exposures and outcomes of interest. Data from administrative health insurance claims and electronic health records are rich sources of information that are used to inform topics ranging from public health surveillance to comparative effectiveness research to identifying patients who may be eligible for inclusion in a specific study or intervention. Data sourced from individual sites can be limited in their scope, coverage, and statistical power. Sharing and pooling data from multiple sites and sources, however, present administrative, governance, and analytic privacy challenges. But in a distributed data network, there's no central repository of data. Rather, data are maintained and reside behind the firewalls of each data contributing partner in a network. They then transform their source data into a common data model and permit access to the data through the use of a standard query approach. Transformation of data to a common data model ensures standardized application, analytic tools, and methods can be applied to them. The types of real-world data sources used in BEST are claims, administrative billing data, and electronic health record data. It is important to remember that BEST utilizes secondary data. This data is patient interaction with the U.S. healthcare system. Besides granular data, EHRs are almost real-time and cover both insured and uninsured patients. However, the main limitations of EHR is that they only cover snapshots of patients' clinical experience. In contrast, administrative claims are excellent in tracking healthcare continuum, regardless of which healthcare systems are used. However, claims data suffer from long processing time and claims delay. Given the pros and cons of different data sources, we usually evaluate the fit for purpose per study. BEST has access to diverse types of data, including large administrative claims data, EHRs, linked EHRs. EHRs, as mentioned before, provide more granular clinical information such as lab results. On average, data lag in BEST databases is about 3 months, but we're making efforts to gain access to fresher data. For example, in Optum's pre-adjudicated claims, the data lag is 2 months. Time for another poll question. What are some unique characteristics of biologics important to biologic surveillance? A, the blood supply is static and does not need to be monitored continuously. B, unlike drugs, vaccines are administered to healthy populations and children, and therefore there's a low threshold for risk of adverse events. C, both A and B, or D, none of the above. B, that's correct. Unlike drugs, vaccines are administered to healthy populations and children. Therefore, there's a very low threshold for the risk of adverse events. We're transitioning to the final section, and now we're going to be looking at how to use real-world data for regulatory work. In order to understand how real-world data is used for regulatory work, we first need to understand how clinical events are captured within these data sources. Today, we're going to focus on 4 coding systems commonly used within BEST data sources. First, ICD codes. ICD codes were created by the World Health Organization as a way of standardizing healthcare classification. ICD codes are diagnostic codes that represent all aspects of medical diagnosis, including symptoms, social circumstances, cause of injury or disease, and more. Next, we're going to focus on what we call HCPCS codes, Healthcare Common Procedural Coding System. And this includes Common Procedural Terminology, CPT codes. HCPCS codes are the most common codes we encounter for biologics because they are used to bill for administration of the biologic, such as vaccines. These codes can be nonspecific. Here's an example of some of the non-specificity that we see. So as you can see, these are some of the HCPCS codes, and in some cases, they correlate with one brand of, in this case, factor VIII. But in some cases, one code correlates to 3 different brands. This is a limitation for conducting biologic surveillance and claims data. Then we go on to NDC codes, National Drug Codes. They're the most specific codes. Every drug, every biologic has an NDC code. NDC codes can specify brands. They can even specify dose. However, for biologics, NDC codes are not always used for billing. Therefore, we're unable to use this coding system for a majority of our surveillance work. And lastly, we have ISBT codes, International Standard for Blood and Transplant. This is a global standard for the terminology, identification, coding, and labeling of medical products of human origin. This includes blood, cell, tissue, milk, and organ products. ISBT codes can only be found in EHR. This is not a billing code, but it is a code that can be used for surveillance purposes. So now that we have the foundation, we understand the types of data and how clinical events are captured, let's get back to the original question, how to use real-world data to support regulatory first. So first we start with the regulatory question. So let's start. First, we're going to need to identify our population of interest. And in this case, we need to think about how do we define that population within the confines of secondary data. Do we need the age in days, weeks, or will months or years be sufficient? Because in some cases, some of our data sources don't have the granularity of days and weeks when it comes to age. Gender, that's pretty straightforward. For the most part, our data captures this kind of information. Race and ethnicity. Our data has a lot of missing information when it comes to race and ethnicity. If this is absolutely necessary for the regulatory question, that would substantially decrease the number of patients that can be assessed. Are we interested in looking at a healthy population or a population with a certain condition? How would we define these conditions? Do codes exist to define a disease of interest? Sometimes we may use presence of a treatment to determine the diagnosis of a condition. Next, we need to discuss the biologic of interest, which is defined as our exposure, or in this case, the intervention. We need to consider if a comparator group is needed. So when we think about the exposure, is it a biologic, like a vaccine or a blood product, or is it a procedure, such as a transfusion? Some of the other things we need to consider is brand. Do we need brand information? Do we need lot information? Again, this will help us determine which data sources can be used. Another question that we have to consider is absence of exposure. This is especially important when it comes to flu vaccines. Because we're using billing data in a lot of these cases, you can imagine that some people don't always go to the doctor to get their flu vaccine. Sometimes they get it at work. And in that case, it would not show up in their claims data. And so if we do not see a flu vaccine in their claims data, it doesn't necessarily mean that they did not get the flu vaccine. Then we need to assess outcome, the adverse event of interest. Is it an acute outcome that would be treated in a clinical setting or can easily be defined using billing code? Or is it a chronic outcome persisting for a long time? One aspect of claims data that is important to remember is that the average time a person has the same insurance is about 18 months to 2 years, which means that I can only follow a patient in one database for about this long. Any outcome that would occur after that, we may be unable to see in our database. In addition, we need to consider the setting of diagnosis. Where is the outcome diagnosed? Did the patient show up at an ER? Were they admitted? All of these different aspects are important in defining the outcome of interest and in determining which database would be the best fit. And last, time. Time is important to understand from different angles. First, there's the unit of analysis of time for the population of interest. If the age is needed, is years sufficient? Would we need the age defined by months or days? We discussed this when it came to the patient population. This pertains mostly to neonates and infants. For the outcome of interest, time is important in defining the onset of the outcome. As we discussed from the previous slide, understanding that there is limited follow-up. Okay, let's walk through an example quickly. This is a test case we used a couple of years ago as we were establishing the BEST initiative. So the question is there a difference in the odds of febrile seizures for children 12 to 23 months of age receiving first dose of MMRV vaccine, MMR and varicella vaccine separately, MMR vaccine without varicella vaccine, and varicella vaccine without MMR vaccine? So first, the patient population. So we know we're dealing with children, and we know that we're dealing with children ages 12 months to 23 months. So we need access to a database that would give us age in months. Also, we're looking for a relatively healthy population, no history of seizures. So now we need to go back into the history of these patients, of these children, and see if there are any codes that would indicate that these children had seizures. Next, we're going to talk about the exposure. So in this case, we're looking at MMRV, no history of MMR or varicella vaccine in their prior look back, meaning that we would look back and make sure that this would be their first time receiving MMR vaccine. And we're comparing this to children that received MMR plus varicella on the same day, so 2 separate vaccines, or an MMR vaccine without varicella, or a varicella vaccine and no MMR. Our outcome of interest is incidence of febrile seizures. This can happen in a medical setting, inpatient, outpatient, or emergency room. And lastly, time. So in this case, we're looking at time in 2 different ways. First, the time period. We were looking at years 2000 to 2017. And then as far as time, we were looking at risk window. So 0 to 28 days after vaccination. So as I'd mentioned, this was a test case. We were testing our system by reproducing components of a published study. This study was by Klein et al. In 2010. Their study objective was to assess the risk of febrile seizures in children receiving first dose of measles, mumps, rubella, and varicella compared to that of MMR and varicella administered separately on the same day. So quickly, we're just going to look at the results. Again, this was just a test case. So here are the results. We looked at slightly different time periods. So in the VSD study, they focused on 2000 to 2008. Our goal was to focus on 2000 to 2017. The limitation of one of the databases that we were using was that it did not go that far back. Another limitation of a database that we were using was that it did not have age in months. So we had to adjust the study to look at children 1 to 2 years of age. It's about the same thing, but slightly different. However, one of the benefits of the database that we were using was that it was very, very large. And so we were looking at almost 1 million children versus the VSD study that only had access to about 83,000 patients that received MMR vaccine. The results were very similar. But again, the point of this was just for us to test the system to see if we could reproduce the components of a study. In summary, the mission of CBER is to ensure that biologic products approved by CBER are safe and efficacious. And today, I hope that I gave you a little insight in how we are fulfilling that mission. If you have any more questions or would like to learn more about the BEST initiative, please visit us at www.bestinitiative.org, and you'll see that we have a list of all the current studies and past studies that have been completed in the last couple of years. We also have access to some of our protocols and final reports. Thank you.
Thank you, Meghna and Joyce, for the very clear and informative talks. We are now at the question and answer portion of the session, and I see that we have some questions in the chat box. Well, let me start with Dr. Alimchandani. So the first question goes to Meghna. Is submission of a pharmacovigilance plan required by FDA? And can a pharmacovigilance plan change over time?
Hi, good morning. This is Meghna Alimchandani. Can you hear me okay?
Yes.
Okay, great. Thanks. So at this time, our regulations do not require submission of a pharmacovigilance plan. However, CBER strongly urges applicants to submit a PVP. So the PVP is a really important aspect of our review. It should summarize your safety concerns for your product and your actions. And it, of course, it can change over time depending on the post-market experience data and data from clinical trials. So it can change. You can submit. It's a living document. So we review many versions of the PVP, and every few years we may ask for an updated PVP when we are doing a post-market safety review. And again, it's not required, but we strongly encourage you to submit a PVP. Thanks.
Thank you. That was helpful. The next question goes to Dr. Abidi. The BEST initiative, which you described, has a number of data sources. How are studies conducted within the multiple databases?
Hi. So how studies are conducted within multiple databases is that we generate one protocol. Also, as mentioned, we have a common data model. So all of our databases convert their data into this common format. And so when a study is conducted, everyone's using a similar format. And so we conduct what we call a federated study. And so each data source conducts the same exact study, and they all use the same protocol. And when we present those results, we don't aggregate it, but we do present them as separate results from each of the data sources.
Great, thank you. Following question is for Dr. Alimchandani. Can the sponsor submit a risk management plan prepared for EMA to the FDA instead of a separate U.S. pharmacovigilance plan?
Yes. So the short answer is yes. It is acceptable to submit the risk management plan format that you have prepared for EMA. However, I want to say that following our FDA review, we may have additional recommendations for safety specifications and PV actions. And sometimes an applicant may choose to have a separate PVP to incorporate our recommendations that may not be applicable to the EU's RMP. So just keep that in mind.
Thank you. Joyce, you're up next. Does BEST have direct access to the data used in studies that are conducted?
So BEST does not have access to the data. Well, I should say FDA does not have access to the data that's used in our studies. And so what happens is each data partner keeps their data behind a firewall. And so what we do is we have our work groups. We work together to generate the specifications for the study. We come up with, what the study question is, and then that's sent to the data partners. They build their programs and they run the studies. They then share only the results with FDA, but not the actual data. So that data is still kept private. And again, we don't have any access to any personal confidential data.
Okay, thank you. Thank you. That makes sense. Next question goes to Meghna. Are sponsors required to submit medication error reports to FDA?
So at this time, our regulations do not require for medication error reports that are not associated with a clinical adverse event to be submitted to our pharmacovigilance databases. But that being said, as part of our routine surveillance processes, we may send information requests to sponsors to provide your summary of certain types of medication error reports. But again, if the medication error report is not associated with an adverse event, it is not required to be submitted to FAERS.
Great, thank you. Dr. Abidi, you mentioned use of an expandable common data model. What do you mean by expandable? And if you can give an example, that would be helpful.
So what I mean by expandable data model is in the past, we've had data models that were very rigid. And so if there were data elements from the medical charts or from the EHR charts that we thought were important to add to our study or for the purposes of our surveillance, it would take years for us to add that to the common data model and then be able to query it. So with the common data model that we have now, it's a common data model that accommodates both claims data and EHR data. So there's a lot of data elements in there. And if there was something that FDA needed access to or information that FDA needed access to, I shouldn't say access, but information that was needed for a study, we would be able to add that data element quickly to the common data model. So one example is the use of ISBT codes. So as I mentioned, ISBT codes are not billing codes. They're actually codes that are used in blood banks and usually only found in EHR data. And in some cases, it's actually separate from the EHR record. So it's only kept in the blood bank in that hospital. And with one of our products that we regulate, which is blood components, regulating the safety of it, we wanted to make sure that we could actually get as granular information for blood transfusions as much as possible. And so with the contractor that we were working with, they were able to add this data element to the common data model. And then each one of our data partners was then able to retrieve that data from the blood bank and then populate the common data model so that studies could be queried. And this happened within a month or two. And it was something that strengthened our surveillance capabilities.
Thank you, Joyce. This is such a good example of how the system can actually work. That's great. Thank you. The next question goes to Meghna. What is meant by the term enhanced pharmacovigilance?
So we use this term occasionally, enhanced pharmacovigilance. So as I told you, routine pharmacovigilance is mandated by 21 CFR 600.80, all the adverse event reporting regulations that we went over. The regulations give us some flexibility to monitor certain adverse events of special interest more closely, and that comes under this umbrella of what is called enhanced pharmacovigilance. So for example, under enhanced pharmacovigilance, we can ask for expedited reporting for adverse events, for a certain type of adverse events, regardless of seriousness or expectedness. So we can tell you, for this particular adverse event of special interests, please submit 15-day reports without regard to whether it was a serious or non-serious report. Similarly, for the periodic safety reports, we can change the reporting interval. So normally under routine PV, the reporting interval would be quarterly for the first 3 years and then annual following that. But for a certain, if we have an open ongoing safety signals, we can ask you to submit periodic safety reports at more frequent intervals. Okay. And then finally, as part of follow-up, you are mandated to conduct follow-up for all 15-day reports, but we can also ask for certain targeted questionnaires to have more streamlined collection of follow-up information. So that all of this falls under this term, enhanced pharmacovigilance. Thanks.
Thank you, Meghna. This is the term that I hear used frequently, so it was very helpful to actually better understand what it is used for and what is meant by it. And we get this question quite, we've received it on a number of occasions from sponsors. So our next question is addressed to Joyce. What are some surveillance requirements that you take into consideration when dealing with biologics?
So some of the requirements that we take into account, so as I mentioned before, there's some unique characteristics of biologics that we have to consider. And we think about the requirements in 3 different categories. So we think about the overall safety surveillance. So when it comes to like newly approved products, this requires near real-time surveillance. And so that means that we need access to faster data and quicker data. And so that's something that we're definitely working on. Also having access to continuous continuous data, during the emergence of any infectious diseases. The second category is the health information. So this includes, making sure that we have, as I mentioned, there's significantly different coding and reimbursement practices when we think about drugs. And so when you think about biologics, making sure that we have access to the HCPCS codes, which we know captures the administration of the biologic, CPT codes, ICD codes. So making sure that our databases are adequately capture that information. And so making sure, so considering access to different multiple sources, blood bank, which I mentioned, registries is something that we're now trying to add to our data sources to make sure that we have access to as much information as possible. And then the population characteristics. So again, we're dealing with healthy populations. So some of the outcomes that we're looking at are very, very rare. And so we want to make sure that we have access to very large data sources so that we can, we can see even the smallest number of cases, and making sure that we can assess high-risk populations such as pregnant persons, the elderly, children, and immunocompromised. Yeah, thank you.
Excellent. Thank you, Joyce. Our next question is for Meghna. Can a sponsor submit the same periodic safety report to different regulatory agencies? For example, can we submit a PSUR prepared for European Medicines Agency to the FDA and fulfill the requirements under the regulations?
Thanks, Larissa. So we get this question a lot. And the short answer is yes. You would need to submit a request for a waiver, so that instead of submitting a periodic adverse experience report or PAIR, which is under our regulations, you are able to submit a periodic safety update report or a periodic benefit risk evaluation report, PIBRUR, which are the European formats for periodic safety reports. So it is perfectly acceptable to do that. You do need to submit a waiver. We also try, if it is feasible, to harmonize the data log points and the reporting intervals to match with other foreign regulatory agencies and reduce the burden for sponsors.
Thank you. Meghna, our next question also goes to you. Does FAIRS have the capability to be searched by a company's name, and then the person who asked the question provides different pros and cons, and if you could respond to the question and also maybe deliberate on the pros and cons of that.
From our, I just wanted to clarify, so there is the public dashboard and there is our access to FAIRS and what we are searching in our pharmacovigilance database. So from our end, of course, yes, we can search by sponsor name. And for the manufacturer reports, we know which sponsors, which manufacturers are submitting it, and we are in constant, communication. If there is any follow-up information that we are looking for a particular case, we will reach out to the sponsor. So all of that information is visible to us on our end. Now, what I don't know is whether you can see this on the public dashboard. So there are other, people who are, much more knowledgeable about sort of the data aspects and what is available on the public dashboard. So I'm not sure if that is available to you to view on the public side, but on our side, rest assured that we are able to see the company names.
Thank you, Meghna. Our next question goes to Joyce. For post-marketing products, does FDA compare adverse events data between a biosimilar and the brand name?
So for the most part, when it comes to biologics, because of the way that the data is captured, we are not able to work on studies comparing brand names unless there's a specific code available for that brand name. And so it's not something that we do often, or I should say we do very rarely. I hope that answers your question.
Thank you. I think it covers it. Hopefully it was sufficiently informative to the person who asked, but if you haven't received the answer that you were looking for or sufficient response, please feel free to send your questions to our Office of Communications in CBER, and we should be able to direct you to the right channels and try to find an answer for you. This applies not to just this session, but generally to any of the sessions that we have today and we had yesterday, and regarding any questions that you may have. So the next question goes to Meghna. What should a sponsor do if they cannot meet the proposed timeline submitted for PMR studies? Because usually dates listed in the approval letter may be more or less estimated times, and what if they change in the future? So what would be a course of action in that case? Thank you.
Hi. So we acknowledge that, of course, this can happen. These are estimated timelines that are listed in the approval letter. There are a couple of things to keep in mind. First of all, it's very important to keep in mind that from the FDA perspective, we determine the study status, for example, whether it's delayed or not, based on the milestone dates that you have listed in that original approval letter. So we are always going to make the determination of the study status based on those original dates. If you cannot meet the proposed timelines, then you would need to submit a justification for why the study is not feasible to be completed as per the original timeline, and propose, revised study milestone dates, and this would come as a good cause request to the agency. So you have to demonstrate good cause for the non-compliance and submit this information to us. Now, the other important point that I want to remind you is that the FDA cannot grant a good cause request unless a study milestone listed in the approval letter was missed. So if your study is not going to be completed by the study completion date that is listed in your approval letter, then you should submit your good cause request close to that date. I would say, not more than 90 days before that date. Don't submit the good cause request to us, months or years before that study milestone, because we will not be able to grant the good cause request until the time that you have missed the milestone. So that's something important to keep in mind. And remember that you are also providing annual updates to us in your PMR annual status reports. So in those reports, you can certainly elaborate on issues that you're having with the study, if there are problems with enrollment or whatever, feasibility issues you're having. And we will be reviewing that as well and providing comments as needed. So I hope that answers that question.
Thank you, Meghna. Thank you. I think this was a very comprehensive answer. I hope it was helpful to the person who asked. Our next question also goes to you, and that is, if a REMS is not required for a new biologic or new drug, does the sponsor have to submit a non-REMS risk management plan as part of the original marketing application?
So this, goes back to the first question that I took: is a pharmacovigilance plan required or not? So a non-REMS PVP, again, it's not required by the regulations, but it is strongly encouraged. CBER strongly urges you to submit a non-REMS, just a standard pharmacovigilance plan for your product.
Thank you, Meghna. And you know what? We have another question for you here. To prepare a risk management SOP, does FDA have an easy-to-follow guideline or examples to follow?
Thank you for this question. So there are multiple FDA guidance documents on the content and the format of a REMS document and the REMS program. And we also have all of the approved REMS programs, CDER and CBER REMS programs that are posted online, publicly available. So these are great examples, and we will be sending you the link for this shortly. So in that link, you can look at multiple REMS programs and how they have been constructed. I also want you to keep in mind that if we make the determination, if the agency makes the determination that a REMS is necessary for your product for the benefits to outweigh the risks, then we will work very closely with you to develop the REMS. I think each REMS program is very unique. So we will be providing you with a lot of, guidance and sending a lot of, language back. There will be a lot of communication back and forth to develop the REMS program. Thanks.
Thank you, Meghna. So switching gears to post-marketing studies, can you clarify difference between PMC and a PMR?
So this is a great question, and we spend a lot of time thinking about when something should be a PMC versus a PMR and vice versa. So the PMR, by its name, it's a post-marketing requirement. It's a requirement. So you are granted approval essentially on the condition that you will conduct the study and complete the post-marketing requirement. And FDA can take certain enforcement actions that are described in the PMR guidance. And I had provided the link in one of the slides. And these enforcement actions may include monetary penalties or misbranding. So again, it's a requirement. And there are enforcement actions associated with it. A post-marketing commitment, on the other hand, is described as a study that is agreed upon between FDA and the applicant. So you would provide annual updates for a PMC just like you would for a PMR. FDA will track the study milestone dates, and we provide, we track both PMRs and PMCs, and the status of the study is posted publicly. So we would do that. However, it's not a requirement. So we do not have any regulatory authority to take enforcement action. So I think that is, the biggest difference.
Thank you, Meghna. Thank you. Thanks to both of you, to Meghna and Joyce. I think we're out of time at this point. I would like to once again thank both of our presenters for their very clear and interesting presentations and for responding to questions. This was such a rich discussion. Thank you. We will now go on break, and after the break, we'll transition to the next session, which will cover the topic of xenotransplantation. The session will be moderated by Dr. Irina Tipper. Thank you. We will see you after the break.
Good morning, everyone. Welcome back from break. I hope you have been enjoying the morning sessions thus far. My name is Irina Tipper, and it's my pleasure to welcome you all to the Xenotransplantation session and welcome the next three speakers, Ms. Judy Archidiakono, Dr. Archana Saddam, and Dr. Patricia Beeston. Judy Archidiakono is a regulatory expert on xenotransplantation products in the Office of Tissues and Advanced Therapies. She also serves as an international policy, a regulatory expert, and lead for the Standards Recognition Program for the Regenerative Medicine Therapies. Prior to joining OTAT Immediate Office of Director in 2007, Judy worked as a research reviewer staff scientist in the OTAT of Division of Cell and Gene Therapies, where she conducted research on human immune responses to porcine cells as a model to study xenograft rejection. In addition to research, Judy conducted regulatory reviews of INDs and IDEs for xenotransplantation products and NK and T cell therapies. Judy holds a BS from West Virginia University and an MS from Clarion University of Pennsylvania. Dr. Archana Saddam serves as a product reviewer in the cell therapies branch within OTAT. At CTB, she provides expert scientific and technical assessments of regulatory submissions for novel cell therapy products to inform scientific and regulatory decisions regarding the safety and effectiveness of cell therapy products. She joined CTB in 2019, and prior to that, she joined the FDA in 2017 as a fellowship with the FDA Commissioner's Fellowship Program. She received MS and PhD in molecular biology and genetics from the University of Delaware in 2017. And finally, Dr. Patricia Beeston has been a medical officer at the Food and Drug Administration for more than 20 years. She began on the Center for Drug Evaluation and Research, evaluating endocrine drug products. She then moved to the Center for Devices and Radiological Health, where she provided clinical consultation on endocrine-related devices, including diagnostic, software, drug delivery, and combination products. She is now in the Division of Clinical Evaluation, Research, and Pharmacology and Toxicology within OTAT, where her review work includes regulatory applications for islet cell transplants, gene therapies for inborn errors metabolism, and xenotransplantation. She is a member of the Xenotransplant Working Group. Dr. Beeston earned a BS in zoology from University of Maryland College Park, a PhD in anatomy and neurobiology, and an MD from Medical College of Pennsylvania in Philadelphia. She completed her residency in internal medicine at the Medical College of Pennsylvania and was a fellow in diabetes, endocrinology, and metabolism at Penn State Hershey, where she did basic research in addition to her clinical responsibilities. And with that, I will pass it over to our speakers.
Thank you for the introduction. I'd also like to thank the organizers for inviting me to speak to you on FDA regulatory oversight for xenotransplantation products. The learning objectives for this session are to define a xenotransplantation product, identify the FDA centers responsible for regulatory oversight of xenotransplantation products, to list the risks associated with xenotransplantation products, and the stages of rejection, as well as measures that can be taken to reduce risks to recipients of xenotransplantation products. I will first define xenotransplantation and then provide a brief history of xenotransplantation policy development and current activities, as well as regulatory expectations for xenotransplantation products and recommendations for patient and animal sample collection. Xenotransplantation is any procedure that involves the transplantation, implantation, or infusion into human recipient of either live cells, tissues, or organs from a non-human animal source, or human body fluids, cells, tissues, or organs that have had ex vivo contact with live non-human animal cells or tissues, and these definitions can be found in the FDA Xenotransplantation Guidance as well as the PHS Guideline on Xenotransplantation. Examples of xenotransplantation products are whole non-human organs, non-human cells or tissues, extracorporeal perfusion of human blood over or through non-human cells or organs, and the administration of human cells previously cultured with non-human cells. What are not xenotransplantation products are biologic products, drugs or medical devices sourced from non-living cells, tissues, or organs. Examples of these include, but are not limited to, porcine insulin and porcine heart valves. I'd just like to point out that porcine heart valves are not xenotransplantation products because they do not contain viable cells. With this very busy slide, I wanted to demonstrate the number of public consultations that took place to formulate current xenotransplantation policy. FDA guidance for industry in 1999 was published on public health issues posed by the use of non-human primate xenografts in humans. Initially, it was thought that non-human primates would be the best donors because of their similarities to humans, but this was during the AIDS epidemic, and so there were concerns about transmission of infectious diseases from the non-human primates to humans. So other animal sources were encouraged. The Public Health Service Guideline on Infectious Disease Issues in Xenotransplantation was published in 2001, and in 2003, FDA published a guidance for industry on product, preclinical, and clinical issues concerning the use of xenotransplantation products in humans. So the PHS guideline and the FDA guidance of 2003 continue to be the primary resources for advice for xenotransplantation product developers. So to continue on the timeline, in 2008, so there was some time that passed between the development of policy, the World Health Organization first consultation on regulatory requirements for xenotransplantation clinical trials took place. And the next year, FDA Advisory Committee meeting on animal models for porcine xenotransplantation products intended to treat type 1 diabetes or acute liver failure took place. And then shortly after that meeting, the International Xenotransplantation Association, or ICSA, published a consensus statement on the conditions for undertaking clinical trials of porcine islet products in type 1 diabetes. More recently, FDA updated their guidance for industry on xenotransplantation, so in 2016. In 2017, FDA and ICSA held a joint workshop on advances in xenotransplantation. The purpose of this workshop was to bring industry up to date on FDA expectations for xenotransplantation, as well as inform FDA on the current research advances in xenotransplantation. In 2018, WHO and ICSA held a joint meeting that included regulatory parties as well as developers of xenotransplantation products and researchers. So this meeting resulted in an international consensus on clinical trials for xenotransplantation. This document aligns with FDA policies and procedures, and at this meeting they also adopted FDA's definition of xenotransplantation product. So most recently, as most of you have probably heard about, a genetically altered pig kidney was transplanted into a human decedent in 2021. In the dark blue box here, I have listed the publication that discusses that experiment. I want to note that FDA does not have oversight for experiments using human decedents, and so this transplantation took place without FDA purview. In 2022, a genetically altered pig heart was transplanted into a human recipient. And here I've also provided the reference that discusses this xenotransplantation. The centers with regulatory oversight for xenotransplantation products depends on the regulated articles. So the Center for Biologics Evaluation and Research, or CBER, is responsible for regulating the animal organ, cell, or tissue to be transplanted. And then follow-up would be on the patient post-transplant, and that would include the function of the transplant and then monitoring for infectious diseases. And I just want to point out that there are more things that will be followed up on, but as opposed to what's followed up in the on the animal side, I just listed these two functions. So for the Center for Veterinary Medicine, or CVM, the regulated article is the animal with an intentional genomic alteration. And the follow-up would be on the stability of the genetic alteration in the herd. At the bottom of this slide, I provided a reference that refers to how the Center for Veterinary Medicine and CBER and other FDA centers work together to evaluate products that come from genetically altered animals. In the next two slides, I'm going to go over the components of CBER's evaluation for xenotransplantation products, and then components of CVM's evaluation. The source herd and source animals are co-reviewed by CBER and CVM. And what we're looking at are appropriate breeding, maintenance of animal health, and maintenance of the animal facility. For the individual source animal that is used for transplantation, procedures should be in place to minimize infectious disease risks, and this includes screening and harvesting and handling of tissue, cells, and organs. Product processing and testing would include process control, so current GMP should be followed as closely as possible. There's also expected product characterization, and this would include safety testing and lot release. Preclinical assessments include extrapolation of cross-species infections, immune reactions between the source animal and the recipient, and the function of the xenotransplantation product. Clinical issues include scrutinizing the protocol and informed consent. Patient selection and the follow-up screening will also be discussed. So I just want to highlight that the product processing and testing, preclinical and clinical assessments these are very short lists, so there are more detailed evaluations that will take place. Components of CVM's review include the product description, which is pretty self-explanatory, and then molecular characterization of the constructs that are used to modify the animal. They will also review the molecular characterization and phenotypic characterization of the animal itself. They will review the claim validation. So what claim validation is if the sponsor claims that, for example, the pig has been knocked out for the alpha-gal sugar, they will have to provide data to show that that is actually the case in the animals. CVM will also review environmental, food, and feed safety issues, as well as the genotypic and phenotypic durability plan. So they will monitor the expression of the genetic modification over time in the animal. As you can imagine, there are many risks associated with the use of xenotransplantation products. I'd first like to focus on the transmission of known and unknown pathogens and point out that we can only screen for what we know. So, as I will discuss later in the presentation, their monitoring and follow-up of patients and then collecting samples from the donor animal are very important. Also, the risk of zoonotic infections to the patient, personal contacts, including healthcare professionals and the general population. Adverse inflammatory and immunological responses by the host to donor cells or secreted molecules is of concern also. These inflammatory reactions also can lead to rejection. And then the physiological and metabolic incompatibility between the animal donor organ and humans. Also, the deleterious effects of the use of high-dose immunosuppressive agents. So since pigs and humans are so distantly related, you can imagine that there is more immunosuppression that will be needed to maintain the transplant. So in the next section, I would like to talk about pigs as source animals for xenotransplantation products. Currently, pigs are the donor of choice for xenotransplantation, and this is because of their similarities with humans regarding the size, function of tissue, cells, and organs. Humans have been exposed to domesticated pigs for centuries, and pigs can be genetically modified to make them more compatible with humans. In this slide, I'd like to discuss xenotransplant rejection. This is quite similar to what happens in allotransplant. So within minutes to hours, hyperacute rejection can take place. And this is hallmarked by activation of complement and the formation of antibodies to the xenograft. Within days, vascular injury can occur, and this is due to coagulation incompatibility, platelet activation, and clot formation. Within days to weeks, a cell-mediated rejection would take place, and this is due to the activity of natural killer cells, T cells, and macrophages. And then within months to years, chronic rejection can take place in response to novel antigens in the graft. And similar to allotransplantation, this would be dealt with by immunosuppressive drugs. In the next two slides, I will provide a list of genetic modifications that have been conducted in pigs, and the details of these can be found in the literature. But I have grouped these according to the function that is being modified. So the first three modifications are deletions of xenoantigens that are found on the surface of the pig cells. For complement regulation, coagulation regulation, anti-inflammatory and anti-apoptotic activity, human genes are knocked into the pigs so that the pigs express these human proteins. In this side, I've also grouped the modifications according to the impact they have on the rejection cascade. So first, the regulation of natural killer cell mediated responses can be modified by the expression of HLA-E, so the knock-in, or knockout of ULBP1. The regulation of macrophage-mediated responses can be modified by CD47 gene expression. T-cell-mediated responses can be modified by the expression of CTLA4 or knocking out SLA class 1. Genetic modifications have also been performed to inactivate porcine endogenous retrovirus and the pig growth hormone receptor. It has been observed in preclinical models of heart transplant that the heart can grow over time after it's been transplanted into the non-human primate. So this can hopefully assist in the growth problem. We must, however, think about the risks associated with genetically altered source animals. For those of you that are familiar with genome editing, we are concerned about off-target effects. And we are not sure the number or the types or combination of genetic modifications that are needed for a most effective control of host responses. Also, off-target effects or even on-target effects could lead to alter organ function, metabolic changes in organs or organ systems. We just don't have information about that right now. And then there is the long-term effects that we're not sure. There's an unknown impact on performance of the organ over time. It is almost impossible to eliminate all the risks associated with xenotransplantation, but with appropriate animal testing, these risks can be mitigated. FDA's goals for source animals used for xenotransplantation include a balanced risk assessment, layers of safety, and the use of best practices and validated technologies. In the next few slides, I'm going to talk about the animals that are used for transplantation with a little bit more detail than what I've provided previously. So animals should be bred from closed herds of known origin, preferably from the United States. So a documented origin infectious disease status is required. 2 or more generations and specific pathogen-free conditions before clinical use is expected. Control of all the animals, including gamete donors. I want to point out that we've had some developers want to source animals from slaughterhouses, and there's really no control here of those animals and where they came from and what they've been exposed to. So they are not appropriate source animals for organ cells or tissues or even gamete donors. Also, the maintenance of animal health is important. This would include screening, sentinel animal testing, and the agents and methods and the frequency of testing must be justified to the agency. Appropriate feed would be feed free of rendered animal materials. It should be natural, and non-sterile materials should be excluded, and appropriate health care for the animals. Maintenance of an appropriate animal facility, also important. They should be kept in a well-controlled and monitored pathogen-free facility with appropriately trained staff with limited access, and these facilities are subject to inspection by FDA. For the individual source animal, procedures should be in place to minimize infectious disease risks, and this includes quarantine prior to use, generally 3 weeks or more before the transplantation is to take place. So this means removal from the rest of the herd. A plan for biosecure transportation should be in place, and that generally means transplantation of the animal or the organ from the animal facility to the clinic. These procedures should take place in well-controlled surgical environments for harvesting cells, tissues, and organs. With regards to screening, a biopsy of the to-be-used tissue is preferred. Infectious disease screening as well as a histopathology should be conducted, and a full necropsy of source animal tissues is recommended. The harvest and handling of the cells, tissues, and organs should be well documented, and there should be an archiving of samples. And that archiving of samples will be discussed later. In the next section, I would like to discuss the collection and harvesting and storage of animal and human samples. And these recommendations are outlined in the PHS guideline on xenotransplantation and then reinforced in the CBER guidance on xenotransplantation. The goals for establishing sample archives include ensuring the health and safety of the patient, the recipient, and their close contacts, and they can provide a source of materials for look back in the case of patient health or public health issues arise. In this slide, I'm describing the samples to be collected and archived, and these may change according to case-by-case situations. For the donor or source animals, the portion of the harvested material, so parts of the cell, tissue, or organ, should be collected, as well as plasma and leukocytes. The timing of the collection should be at predetermined intervals prior to harvest, at the time of harvest, and then post-mortem. For the human recipient or the patient, blood, plasma, saliva, and leukocytes should be collected pre-transplant, post-transplant, and at predetermined intervals and post-mortem. How will the samples be used? Samples for the public health service, so should a public health issue arise. A sample or samples for the recipient diagnosis and care. Samples for use by the sponsor, so maybe research, and then samples for use by the FDA. These samples should be labeled and cataloged in a manner allowing for the linkage between the patient samples and donor animal samples. The samples should be stored in media appropriate for RNA, DNA, cell viability, and antibody preservation. There should be a backup plan in case the company goes out of business, and the herd records and patient samples should be stored for 50 years. In the next two slides, I'd like to provide some challenging questions that I hope the answers have been clarified in my presentation. So the first question, which of the following are not examples of xenotransplantation products? A, pig kidney transplant into human recipients. B, pig heart valve implantation. C, human stem cells cultured on mouse feeder layers. Or D, pig epidermis to treat burns in humans. So imagine the Jeopardy music going on. And now I will provide the answer, and it is pig heart valve implantation. As you may recall, I stated that pig heart valves do not contain viable cells, and therefore they do not meet the definition of a xenotransplantation product. The next question, which of the following measures can be taken to reduce risks to recipients of xenotransplantation products? A, source pigs from closed herds. B, genetically modify pigs. C, source pigs from slaughterhouses. D, screen pigs for infectious diseases. A, B, and C. Or A, B, and D. The Jeopardy music is now playing. And the answer is A, B, and D. As I noted, source pigs from slaughterhouses are not appropriate. I would just like to summarize by saying that the use of appropriate source animals with multiple levels of safety can reduce the risk of zoonotic infections in humans. Genetic alterations in pigs may prevent rejection and reduce the transmission of infectious diseases. Collecting and archiving cells and tissues from source animals can protect the health and safety of the patient and the public. And here are some resources that you can look at to further delve into the information that I present. And thank you so much for your attention. I will take questions.
Good afternoon. My name is Archana Siddham. I'm a product reviewer at Cell Therapies branch at the Division of Cell and Gene Therapies at OTAT. Today I'm going to talk about the chemistry manufacturing controls or CMC considerations for xenotransplantation products. Before getting into the talk, in the next 2 slides, I'll go over the learning objectives and provide an outline for today's presentation. The learning objectives are to gain an understanding on the FDA centers involved and the regulatory oversight in review of xenotransplantation products, and understanding the types of xenotransplantation products. And the last one is to get familiarized with CMC considerations for manufacturing of xenotransplantation products, such as product characterization and safety testing of these products. Next, here is the outline for today's presentation. First, I'm going to provide a brief description on what are xenotransplantation products and different types of xenotransplantation products. And finally, in the rest of the talk, I'll go over the testing recommendations and expectations, such as testing for identity, purity, potency, and safety for these xenotransplantation products. In the previous talk, our CBER Xenotransplantation Product Policy expert, Julie Archidiacono, already introduced you to the definition of xenotransplantation, and I'm also going to repeat this information quickly once again. As described in 2016 FDA guidance, xenotransplantation is defined as any procedure that involves the transplantation, implantation, or infusion into a human recipient of either live cells, tissues, or organs from a non-human animal source, or human body fluids, cells, tissues, or organs that have had ex vivo contact with live non-human animal cells, tissues, or organs. At present, there are 3 types of xenotransplantation products. First one, whole non-human organs for transplantation into human recipients. Second, non-human cells or tissues for implantation into human recipients. And the last type is products that are considered xenoproducts due to their contact with animal cells, tissues, or organs. For example, extracorporeal perfusion of human blood to non-human cells, and other example can be human cells cultured ex vivo with non-human cells and administered to human recipients. As detailed in the reference Staff Manual Guidance 4102, Center for Veterinary Medicine, or CVM, and CBER have regulatory oversight on xenotransplantation products. CBER regulates xenoproducts, that is, animal cell, tissues, and organs that are developed for clinical use, and patient follow-up post-transplantation, that is, monitoring the transplant function, whether it is functioning as it's supposed to, and monitoring potential transmission of infectious agents post-transplantation. And for CVM, the regulatory article is the animal that contains intentional genomic alterations. And CVM also has regulatory oversight on the follow-up on the stability of the genetic alteration in the animal herd from which the donor animal is derived. Now, let's look at the review teams across FDA centers that get involved in review of the xenotransplantation products depending on the product type. At CBER, xenoproducts are regulated at OTAT, and the standard OTAT review team includes regulatory project managers, reviewers from CMC, pharmacology and toxicology, and virology disciplines, and medical officers. And the extended review teams within CBER includes staff from various disciplines such as veterinary medicine, compliance, stats and epidemiology, and manufacturing. The review of some xenotransplantation products require a collaborative review with other centers, and for this we reach out to reviewers in other centers for feedback and help in the review process. For example, for review of GE animals, reviewers from Center for Veterinary Medicine are consulted, and for combination products, that contain both xenotransplantation product and device. The device component, for this device component, consultants from CDRH will be involved. And also we would reach out to CDER consultants for help with review of drugs, for example, for a monoclonal antibody that is used in the manufacture of Xeno product. And for policy-related issues, we would reach out to policy experts. And in addition to conducting collaborative review with other centers within FDA, sometimes, depending on the product, outside scientific and medical experts, patient advocates, and ethicists might get involved. Components that are part of CBER evaluation include source herd, source animal, product processing and testing, preclinical assessment, and clinical issues associated with the Xeno product. Our previous speaker, Judy, talked about the source herd and source animal in her talk, and our next speaker, Dr. Patricia Beeston, will talk about approaches to clinical development of xenotransplantation products. And in my talk, I'll focus on xenotransplantation product processing and testing that includes manufacturing process controls, product characterization, safety testing, and product release testing. Before talking about manufacturing considerations, let's look at the types of Xeno products. There are 3 types of products: whole organ from source animals, for example, heart and kidney from genetically altered pig; non-human cells and tissues, such as islet cells derived from pigs; and the last one, combination products that contain Xeno product and device. For example, as shown here, extracorporeal perfusion of human blood in patients with liver failure, and using this device in which the porcine hepatocytes are positioned outside of a hollow fiber device, the patient blood and plasma will perfuse through this fiber lumen. And I'm going to talk about CMC considerations for these products in next slides. So now I'll talk about manufacturing and process controls recommendations that can be followed to minimize potential contamination during manufacturing and also minimize cross-contamination between lots. Manufacturing process controls should be in accordance with the CGMP regulations that are described in 21 CFR Part 210 and 211. Facilities to be used should be designed to minimize potential contamination of harvested and processed cell tissues or organs, and also to minimize cross-contamination between lots of these cells, tissues, and organs. Some examples of these precautions to consider to prevent contamination are outlined here, such as following aseptic techniques during harvesting of cells, tissues, and organs, controlled procedures to avoid introduction of infectious agents during harvesting, keeping all product contacting surfaces and equipment sterile and pyrogen free by incorporating stringent cleaning and sanitization schedules, and taking precautions to avoid potential surface contaminants carryover during transfer of animals from the animal facilities to the harvesting area, and establishing environmental monitoring to monitor the quality of harvesting and processing areas. And the last one, another important point to consider, is inactivation or removal of adventitious agents. In this part of the talk, I'll go over the xenotransplantation product characterization recommendations and will provide some details on identity, purity, potency, and safety testing. Testing requirements for Xeno products are same as for the other biologics, as most of these products are regulated as biological products, and that means testing the xenotransplantation products also for potency, safety, purity, and identity when applicable. 21 CFR Part 610 describes types of assays that are required for licensed products, and the sponsors should use similar tests during investigational stages of product development. Safety testing should be designed to detect infectious agents and other microbiological contaminants during processing and in the final product. Product testing should include verification of either inactivation or removal of adventitious agents, and the approaches for meeting the above-mentioned testing requirements will depend on the type of the Xeno product. But let's look at the testing recommendations for whole organs. For xenoproducts such as pig kidney and heart that are transplanted immediately after removing them from the source animal, it may not be possible to conduct all tests on the product and have all test results available before transplantation. In these situations, it is recommended to conduct source animal testing prior to organ harvest. For test samples, organ biopsy or relevant surrogate sample such as adjacent tissues or contralateral organs can be used to conduct safety and identity testing. Though test results might not be available before transplantation, assays should be completed and results should be available and recorded in the certificate of analysis. And as part of the potency testing of whole organs, function of the organ can be evaluated, such as measuring hormone secretion, mechanical function, evaluation of blood pressure before the harvest. Next one, for the cells and tissues that are stored, processed, testing for safety, identity, purity, and potency should be included. And unlike the whole organs, when possible, these testing results should be available and recorded in the certificate of analysis before transplantation and should be used as part of product release testing. Additionally, this testing scheme is also applicable to xenoproducts that contain human cells that came in contact with cells or tissues of non-human cells, such as co-culturing of human cells with non-human cells or tissues. In next few slides, I'll go over this testing in more detail. Similar to other biologics, for identity testing, suitable assays should be developed to assess the identity of active components in cells and tissues that are processed and or stored. As part of the identity testing, relevant cell and tissue types can be identified using various techniques such as immunological, immunohistological, biochemical markers, and also when applicable, histological evaluation can be used. Next, depending on the manufacturing process, for example, when animal facility handles more than one strain or species of animals, verification of the identity of strain and species is important and can be included as part of this identity testing. For the purity testing, for heterogeneous products such as products that contain multiple types of cells or cellular implant that contain unrelated tissues and cells that are not completely removed during tissue processing or in ex vivo culture, determination of the purity of the product is important. Quantitative methods should be developed to assess the presence of active cell type as well as contaminating cell type in the final product. Various techniques such as morphological, molecular, and biochemical based techniques can be used to test for the presence of both active and contaminating cell types. For human cells that had ex vivo contact with non-human cells or tissues, testing to assess the presence of non-human cells in the final product should be included as part of the purity testing. As the assessment of purity of xenoproduct is an important point in ensuring manufacturing consistency, if possible, purity testing should be included as part of lot release testing. Also, for xenoproducts that contain purified population of cells, such as an established cell line, the final product should be tested for purity. And next, endotoxin testing in the context of product purity will be discussed in the next slide. For products that are cultured and stored, endotoxin testing is required. Now, the type of endotoxin assay and assay sensitivity and specificity should be provided in regulatory submissions. During product developmental stages, bacterial endotoxin testing can be performed in lieu of rabbit pyrogen test. However, if endotoxin assay is used after licensure, equivalency of endotoxin assay with rabbit pyrogen test should be demonstrated at or before the time of licensure application. Once again, similar to other biologics, potency assays should be performed to measure intended biological activity of the xenoproduct. For example, potency assays can be developed to measure various molecules secreted by the specific xenotransplantation product, such as inflammatory cytokines, hormones, and also neurotransmitters, for example. And when relevant, development of suitable potency assays should proceed with product development. In addition to the chosen potency assay to measure the biological activity of the xenotransplantation product, cell viability can also be included as part of the potency matrix. In the next few slides, I will talk about safety considerations in xenotransplantation product manufacturing. That includes microbiological and infectious disease testing and viral inactivation. Safety testing of xenotransplantation products should include sterility testing for bacterial and fungal contamination, testing for mycoplasma, and for viruses. Test methods used to detect these infectious agents should be qualified during product development stage and should be validated for licensure application. For bacteria, fungi, and mycoplasma, alternative test methods to the ones described in 21 CFR Part 610 can be used. However, the sponsor should provide data on the sensitivity, specificity, and reproducibility of these test methods to support the use of alternative methods. The selected infectious agents and the test selected should be appropriate to animal source, including species, strain, geographical origin, and the type of tissues, cells, and organ to be used. Next, I'll go over some recommended testing approaches for detection of viruses. First approach is to detect potential viral contaminants by using culture assays. For this, cells from xeno products should be co-cultured with a panel of appropriate indicator cells to amplify potential viral contaminants. And the panel of cells used in these co-culture assays should include a cell line that is representative of source animal species and a cell line that is representative of tissue type that is used in the manufacture of that particular xenotransplantation product, and a human cell line. These co-cultures should be observed routinely for any unexpected changes, for example, observing for cytopathic effects, reverse transcriptase activity, and visualization of these co-cultures by electron microscopy to identify potential morphological changes and to recognize presence of certain types of viruses. Next, testing for activation of latent viruses that can occur due to either immunosuppression or transplantation. Additionally, ex vivo culturing of cells can also activate latent viruses. Suitable testing methods to detect latent viruses depend on the tissue type and the virus in question. And sponsors should determine a suitable testing method based on the xenotransplantation product. Identification of latent viruses can be useful in terms of monitoring the transplant recipient for the presence of those viruses. And the last one, for the detection of certain viruses that may not be found by in vitro culture methods, appropriate in vivo assays should be used. Next, as we know, there is potential risk of transmission of zoonotic infections with xenotransplantation products. And in this slide, I'll talk about zoonotic viruses found in pigs that can potentially infect transplant recipients on contacts. And the first one is porcine endogenous retroviruses, or PERVs. There are three types of PERVs, PERV A, B, and C. PERV A and B are present in the genomes of all pig strains, and PERV C is integrated into the genome of many, but not all pigs. Two of the PERV subtypes, that is PERV A and B, can infect human cells, and therefore PERVs pose a risk when pig cells, tissues, or organs are transplanted to humans. So as mentioned, PERV A and B are able to infect human cells, and PERV C is restricted to pig cells only. However, PERV AC recombinants can infect human cells. And the next virus is porcine sarcovirus 3, or PCV3. That is a recently described virus. And has been shown to cause porcine dermatitis and nephropathy syndrome, or PDNS, multisystemic inflammation, and reproductive failure in pigs. And potential transmission of PCV3 to humans is currently being studied. And the other virus is porcine lymphotropic herpes virus 1, or PLHV1, that is closely related to Epstein-Barr virus, or EBV, that causes severe disease in humans. PLHV1 has been shown to cause a post-transplantation lymphoproliferative disorder, or PTLD, in many pigs that is similar to human PTLD that is observed in recipients post-transplantation. Though there is no evidence of PLHV1 transmission to humans, it is important to use PLHV-free donor pigs in order to achieve safe xenotransplantation. In this slide, I'm going to take PERV as an example and talk about some assays that are available to detect PERVs. However, it is the sponsor's responsibility to adopt suitable testing methods based on their product. Both direct and indirect methods can be used to detect PERVs, and some of the direct methods are PCR based and listed here, such as RT-PCR, qPCR, ddPCR, and also other techniques such as electron microscopy, Western blotting of cells or viral lysates. Immunofluorescence can also be used as part of the direct methods. And indirect methods that can be used for PERV detection are Western blotting and ELISA-based assays. As mentioned in previous slides, xenotransplantation products should be tested by co-culture with appropriate indicator cells to amplify infectious retroviruses. And it is important to know that evidence for virus protection is not necessarily indicate that the xenotransplantation product is unsuitable for clinical use. Now, let's look at the testing recommendations for combination product. As mentioned before, combination products contain both xenotransplantation product and device. As described in the previous slide, extracorporeal bioartificial liver is considered as a combination product, for example. Combination product manufacturing includes both cellular component manufacturing and device manufacturing. And as described in the previous slide, cellular component of the combination product, that is the xeno product in this context, needs to be tested for safety, identity, purity, and potency. And then for the device, as the device design can impact the functionality of the cellular product, proper testing needs to be conducted for device components to assess the device performance. And the final combination product should be tested for parameters such as device integrity and biologic device compatibility. For certain combination products containing xeno product and device, physical barriers are used to separate the xeno product from human tissues and therefore can potentially prevent or minimize the risk of transmission of certain infectious agents. In these scenarios, where physical barriers are used to reduce the risk of infectious agents transmission, the sponsors should conduct validation studies and submit data to demonstrate inhibition of specific infectious agents transmission, and also should demonstrate the barrier integrity. Physical barriers can also be used to protect the xeno products from the host immune system. For this, relevant studies such as in vitro and in vivo animal studies should be conducted to demonstrate effectiveness of this barrier system in protecting xeno product and also to assess the integrity of the barrier. And we wanted to take certain parameters into considerations when conducting these studies, such as normal physiologic use conditions for the combination product and the conditions under which combination product is exposed to physical and biologic stress, and selecting relevant infectious agents for testing the barriers, such as using infectious agents that are potentially present and relevant to that particular xenotransplantation product. And last, use of agents that can demonstrate physical properties of the barrier, such as its permeability to viruses and other particles with different properties, such as size, shape, and charge, etc. With this, this concludes my presentation on the manufacturing considerations for xenotransplantation products. And next, let's take a look at a couple of challenge questions. For these challenge questions, I'll give everyone a few seconds to respond to these questions. And the first question is, what are the currently existing types of xenotransplantation products? And the options we have here are whole human organs, animal cells or tissues that are stored, processed, or ex vivo expanded, and combination products containing cells from or in contact with non-human sources and a device, and D, all of the above. I'll give you a few seconds to respond. And the answer is D, all of the above. And the second challenge question is, which of the following is considered as part of the safety testing of a xenotransplantation product? And the options we have here is genotyping, evaluation of presence of active cell types, testing for porcine endogenous retroviruses or PERVs, D, none of the above. And again, I'll give you a couple of seconds for you to respond. And the correct answer is C, that is testing for PERVs. With that, to summarize today's presentation, manufacturing process controls, product characterization, and safety testing are some of the important CMC considerations for xenotransplantation products. And manufacturing requirements described in 21 CFR Parts 210 and 211 should be met for the manufacture of these products. And as for the other biologics, xenotransplantation product characterization should include testing for safety, identity, purity, and potency when applicable. And lastly, selected infectious agents as part of safety testing should be appropriate to the source animal species, geographic origin, and the cells, tissues, or organs. And here are some resources for xenotransplantation products for your reference, for your review. And here is the contact information, and thank you.
Good morning. I am Patricia Beeston, a clinical reviewer in the Office of Tissue and Advanced Therapies, Center for Biologics Evaluation and Research. Today I will be discussing clinical review considerations for xenotransplantation. The learning objectives for today are to describe how chemistry and manufacturing controls, or CMC, and pharmacology and toxicology, or PT, data are considered when reviewing a clinical study or protocol. To describe the considerations for benefit-risk assessment and describe the regulatory pathways to the clinic. The presentation will start with a brief overview of transplantation, a discussion of xenotransplantation products, preclinical data considerations, and clinical considerations. With improvements in surgical technique and tools, donor-recipient matching, and immunosuppressive regimens, the success of transplantation can exceed 90% at 1 year, and 10-year survival has surpassed 50%. With the success of kidney transplant being greater than that for liver, which is greater than that for heart. And living donor transplants are more successful than when a cadaveric donor is used. While these are life-saving and life-improving strides, there is a shortage of donors, deceased or living, compared to the number of patients on waiting lists, and some potential recipients have characteristics that make achieving a match near impossible. To address the imbalance of the need for transplantation and the availability of donors, the use of organs from other species has been considered for more than a century, with tissues and cells being investigated in the more recent past. As discussed previously by Ms. Archidiakonou, there has been much interest in the considerations for donor animal development, the requirements for immunosuppression, and the risk of zoonoses. We must remember that the purpose of transplantation is to provide replacement of function for organs, tissues, or cells that are no longer able to support life or treat serious and life-threatening conditions in patients. Therefore, it is important to consider if the product obtained from the source animal is sufficient to approximate the physiology of the human organ, tissues, or cells that is meant to replace. Surgical techniques for organ transplantation, heart, lung, liver, and kidney are well established. However, there are no data to determine what criteria for organ selection, such as the age of the source pig or the size of the organ, are appropriate. As presented previously, the range of xeno products extends beyond whole organs. Today's brief presentation will be limited to products derived from swine or pigs. While no specific products will be discussed, we will use xeno kidney transplantation in examples. Additionally, we will limit discussion to transplants in adult subjects or patients. The clinical review starts with the input provided by the CMC and PT reviewers, as this information forms the basis for the evaluation of the safety mitigations contained within the proposed clinical protocol. As presented by Dr. Sidem, the CMC reviewer determines that the organ, tissues, or cells obtained from the source animal meets the requirements for transplantation. The PT reviewer if the animal model is appropriate for the clinical condition or disease. These considerations include, but are not limited to, the route of administration, which should mimic the proposed clinical route as much as possible and include the surgical approach, delivery devices, concomitant medications, and immunosuppressive regimens that would be the same or similar as that proposed for the clinical study. While immunosuppressive regimens for allogenic transplants are well established, immunosuppressive regimens that are appropriate for the xeno organ, tissue, or cells are not. PT evaluation of immunosuppressive regimens for xenotransplantation in non-human primates is limited because the commonly used drugs are not effective or well tolerated by the non-human primate. This also limits the ability to demonstrate prolonged function of the transplanted organ. Again, data gathered from study endpoints for safety and function in preclinical studies will be used to assess the strategies in the proposed clinical studies. An investigational new drug application, or IND, is required prior to initiating a clinical study in the United States for any xeno product. We refer sponsors to two guidances to help with planning the transition from preclinical studies to clinical studies. A link to the CBER website that has links to additional guidance documents is available at the end of this talk. We also refer sponsors to the FDA guidance for xenotransplantation. FDA considers benefit risk at each step of clinical development. The hoped-for benefit is for the transplanted organ, tissues, or cells to provide the intended replacement. However, with this benefit comes many risks, known and unknown. The risks from the route of administration include those from the procedure for implantation, such as bleeding and infection, and the site of implantation, based on the organ, tissues, or cells to be transplanted. Next, let us consider the kidney as an example of an organ for xenotransplantation. In general, if the transplanted organ, tissues, or cells cannot meet or approximate replacement of the human organ, tissues, or cells, this mismatch can pose a risk to the recipient. Allogenic kidney transplant has the expectation that the donor kidney will provide replacement therapy. The move to xenotransplantation requires consideration of the kidney's functions and the need to explore if the xeno kidney can provide replacement for all of these functions. And if not, can the risks of these physiologic mismatches be mitigated? In addition to waste removal, the kidney regulates electrolytes and is a complex organ that produces, converts, and responds to hormones. The actions of these hormones is not always conserved across species. I will provide a few examples of these complex functions. For fluid balance, blood pressure and electrolyte balance, potassium and phosphate wasting has been reported in a pig to cynomolgus monkey bilateral nephrectomy model. Free water wasting has been reported in a non-human primate model and raises concerns for a mismatch for response to vasopressin. In sodium regulation, salt excretion is influenced by several natriuretic peptides which act on the kidney, and salt sparing is achieved through renal sympathetic nervous system and renin-angiotensin-aldosterone axis. Renin is produced by the juxtaglomerular cells within the kidney, and we know that porcine renin does not cleave human angiotensinogen. The vitamin D parathyroid or PTH axis is critical in the maintenance of calcium and phosphate within the appropriate physiologic range. The kidney is the site of 1-alpha hydroxylation of 25 vitamin D to form the active hormone. PTH promotes tubular reabsorption of calcium while inhibiting phosphate reabsorption. The amino acid sequence for PTH is not conserved between humans and pigs, and the response of the pig kidney to human PTH has not been described. Erythropoietin is important in stimulating red cell production. We know that porcine erythropoietin is 80% homologous to non-human primates, and it does not support non-human primate erythropoiesis. We also know that porcine erythropoietin does not support human erythropoiesis. Exogenous drugs. We must also consider the pharmacokinetics and pharmacodynamics of drugs that will be used in the peri-transplant period to provide immunosuppression and to manage the recipient's other medical problems or complications that may occur from the transplant procedure or immunosuppression. There are drugs that act on the kidney. It is important to understand if the response of the xeno kidney is similar to that of the human kidney. For example, the SGLT2 inhibitors used in patients with type 2 diabetes. The kidney can act on some drugs. If the metabolism of the drug, especially a narrow therapeutic drug, is different, this difference could result in underdosing and ineffective therapy or overdosing and possible toxicity. Additionally, some drugs can be toxic to organs. It is important that the drugs used in the post-transplant period are not toxic to the transplanted organ. While not unique to the kidney, it should be noted that there is a mismatch in the coagulation cascade between pigs and primates, which can increase the risk of thrombus formation and requires consideration for the transplant and post-transplant periods. Immunologic risks include cell and antibody-mediated rejection. As stated previously, immunosuppressive strategies for xenotransplantation are being explored. Xenoimmune-mediated glomerulonephritis can result in failure of the transplant. Porcine proteins produced by the kidney can result in immune responses to porcine proteins such as renin and erythropoietin that could result in neutralizing antibodies that affect the analogous human proteins. Additionally, it is unknown if antibody formation may occur that could limit the ability of the subject or patient to subsequently match for an allogenic human kidney or allow for transplantation of an additional xeno kidney. The risks of infections in immunocompromised patients is well described. However, the introduction of organs, tissues, or cells from another species raises risks of new infections. As described by Ms. Archidiakonou, breeding programs and herd management are used for mitigating risks of zoonoses. This process is under the purview of CVM. However, once transplanted, we must consider the risk of zoonotic infections not only in the subject or patient, but also the public. A brief summary of these risks is provided. There is potential for zoonotic infections to adversely affect the subject or patient, especially in the face of chronic immunosuppression. Even if the infection does not enter the host cells, if the pig virus has tropism for the transplanted organ, the virus could damage the organ in the immunosuppressed host, decreasing or negating the benefit of the transplant. Therefore, developers are encouraged to have a seamless mitigation strategy from the evaluation of the herd to harvesting of the pig organ to transplantation and then to post-transplantation surveillance. The surveillance plan should include consideration not only for the subject or patient, but also the contacts as appropriate. Developers are also encouraged to consider the possibility of recombination and emergence of new pathogens that may threaten human or animal populations and the possibility of transmission of antibiotic-resistant plasmids in their risk mitigation plans. When developing a clinical protocol, it is recommended that the sponsor provide a rationale for the proposed therapy, identify an at-risk population, recommend a safe starting dose levels and dose escalation schemes for the patient population. For dosing, the sponsor is encouraged to describe the characteristics of the organ, tissues, or cells that would make it appropriate for the intended recipient. For example, as stated before, there are no data to determine what criteria for organ selection, such as age of the source pig or size of the organ, are appropriate. Preliminary benefit risk assessments are important to identify the expected benefit, the expected risks, and the mitigation strategies that will be used to address those risks. Also, a careful identification of parameters for clinical monitoring should be provided. We will examine these topics a little more in the next few slides. When developing criteria for subject or patient selections, considerations include patients with serious or life-threatening diseases with no other therapeutic options, patients who have the potential for significant benefit, bridge versus destination therapy, availability of rescue therapy in case the transplant fails, patients able to comply with public health measures stated in the protocol, including long-term follow-up, and risks and benefits to the patients and public health. To address the potential safety issues for xenotransplantation, staggering of subjects should at first be conservative. Stopping criteria should be included within the protocol. The schedule of evaluations for immune rejection are defined for allogenic transplantation. However, sponsors are encouraged to consider if alternate or additional testing is required. The post-transplant laboratory imaging study requirements and assessment schedules should be designed to identify possible metabolic or physiologic mismatches, and a plan for management of such mismatches should be in place. Additionally, because the source pigs may have one or more genetic modifications, a plan for monitoring for unintended consequences from these modifications should also be in place, such as for detection of possible benign or malignant tumor formation. As discussed by Ms. Archidiakonou, the protocol should include a schedule for monitoring for infectious agents and a plan for surveillance for new pathogens and new infectious syndromes. Qualified assays should be in place for testing the source pig prior to transplantation and for post-transplantation monitoring. It is important that the recipient agree to a post-mortem examination as part of informed consent. We must remember to consider the uncertainty in our plans and assessment of adverse events. There are many regulatory requirements that need to be met for the submission of an IND to be successful. We encourage sponsors to carefully consider these requirements and related guidances when developing their protocols and compiling the CMC and preclinical data that will be needed to support the IND submission. To facilitate program developments, FDA offers the opportunities for interactions. Interact used to be referred to as pre-IND. It is important to understand that only one Interact and only one pre-IND meeting are provided. Balance between late interactions and those that are too early should be considered when asking for meetings and preparing briefing documents. Pre-IND should be requested prior to the conduct of definitive preclinical studies. We remind you that meetings are non-binding and we provide the best advice at the time that the meeting is held. This slide contains links to guidances that describe how to request a meeting with the FDA. In summary, decisions to permit first-in-human trials of porcine cellular products are based on established regulatory principles for early-phase studies of all cellular products. There's a need for demonstration of proof of concept in preclinical studies. Evaluation of potential benefits and risks is specific to each product and each clinical indication. Sponsor investigators are encouraged to discuss their programs with the FDA early in development. Our first challenge question, which of the following are potential risks associated with xenotransplantation? Physiologic mismatch, neutralizing antibodies to analogous human proteins, increased risk of rejection, zoonosis, or all of the above? And the answer is all of the above. Our second challenge question, well-developed preclinical assessments, CMC and PT, are required to provide the data necessary to support first-in-human studies and risk mitigation strategies. True or false? And that is true. This slide contains links to the OTAT Learn webinar series and the CBER website, which can provide additional links to guidance documents and other important information. Thank you for your attention.
Thank you so much to our speakers for really informative and clear presentations, and thank you to the audience for your participation. We received quite a few questions, so we will dive in with the very first question for Judy. Will FDA be holding public meetings to discuss the potential clinical use of xenotransplantation products?
Yes, we will be having an advisory committee meeting on June 29th and June 30th. So to get more information on that meeting, I suggest that you Google FDA Xeno Advisory Committee meeting, and it will take you to the FDA landing page that will provide you information on that meeting. Thank you.
Thank you, Judy. That is certainly something to put on our calendars. The next question is for Archana. What are the CMC expectations on the selection of assays for the detection of infectious agents in xenotransplantation products?
Thank you, Irina, for that question. So the expectations here is the selection of assays should be based on the source of the non-human animal live cells, tissues, or organs, and the intended clinical application for that particular product. And assays used for the detection of these infectious agents should have well-defined sensitivity, specificity, and reproducibility related information. And additionally, in the regulatory submissions, the sponsors should provide a scientific rationale to support the selection of these assays.
Thank you so much. Next question is for Patricia. Can studies using organs from one genetically modified pig be used to support studies that would use other pigs with other genetic modifications?
Thanks for the question. It cannot be assumed that organs, tissues, or cells obtained from one genetically modified pig will be representative of those obtained from pigs with other genetic modifications. Other similar products can be used to help support safety and activity. However, they cannot replace well-designed definitive studies using the intended clinical product. Furthermore, it cannot be assumed that the genetic modifications in one pig would produce multiple organs that would be suitable for xenotransplantation. For example, just because a particular pig has modifications that would be appropriate for one organ, for instance, a xeno heart, That does not necessarily mean that the same pig would have appropriate modifications for a different organ source for xenotransplantation, e.g. the kidney.
Thank you so much for that response and providing a clear example. Let's loop back to Judy. Since CVM and CBER both have roles in reviewing xenotransplantation products that come from genetically altered animals, which center should I contact first?
It's preferable that you contact the Center for Veterinary Medicine first, as they will be evaluating the animal. So that's the first hurdle to starting a clinical trial. And it's important to note that CBER and CVM will work together closely when there are meetings that discuss product development for xenotransplantation products. So if they have if you schedule a meeting with them, they will invite us. If you schedule a meeting with CBER, then we would invite CVM. So we want to make sure that we are on the same page as far as what our expectations are for the animal. Thank you.
Thank you so much, Judy. Let's move on to Archana. Can you elaborate on the precautions that need to be taken while harvesting non-human cells, tissues, and organs that is used in the manufacture of xenotransplantation products?
Yeah, thanks, Irina, for again the great question. So some of the precautions that the sponsors can take is using qualified procedures and having qualified procedures in place to prevent the introduction of infectious agents during the harvesting. And Judy talked about this also in her talk, and I'm going to highlight here again that quarantining of the animal prior to use, and also as transportation of source animals sometimes potentially can expose them to the risks that are not usually encountered in closed herds when transporting these animals, maybe using barriers to maintain, to ensure that the source animal contamination is not happening during this transportation. So these will be some of the considerations to keep in mind.
Yeah, thank you so much. Sounds like there are quite a few logistics to think of. Let's move on to Patricia. Do we need to evaluate the entire pharmacopoeia for pharmacokinetics and pharmacodynamics?
The pragmatic approach would be to begin with a study of those drugs that will be required for the procedure and immunosuppression, and those drugs that are commonly used in the perioperative period and to manage complications of immunosuppression. Additionally, because most transplant patients are likely to have comorbidities, it would be important to study the commonly used drugs such as antihypertensives and diabetic medications. It is recommended that studies are comprehensive. For example, evaluating the contribution of hepatic and/or renal metabolism of the drug or toxicities. As if the initial goal is to transplant the kidney, the information could be useful in future studies evaluating the potential for liver transplant. If the xenoheart is the target organ for transplant, the sponsors are encouraged to consider the range of drugs that may be required and determine if the cardiac response to these drugs, especially neurotherapeutic drugs, is as expected. If not, this can be addressed with dose modification, or possibly alternative drugs would be required. The goal is to have enough information prior to a first-in-human study to design a study such that risks can be mitigated and subjects have a favorable benefit-risk profile.
Thank you so much. That was a very clear answer. The next question is for Judy, and it's related to the previous question regarding CVM and CBER collaboration. The audience member asks, if my product is co-cultured with animal cells, do I need to have an interaction with CVM prior to discussing my product with CBER?
CVM only needs to be included in product discussions when a intentionally genetically altered animal is used. So we may consult with them if we have questions about the animal husbandry for that cell, but you would not be expected to talk to them first. And it's important to note that whichever center you contact first, they will be sure to guide you to what who the contacts are for the other center. So there's no harm in contacting either center. Thanks.
Thank you, Judy. Archana, is use of defined media with animal proteins and cytokines, etc., considered xeno?
No, they are not considered xeno products. However, Irina, I wanted to highlight that when using these reagents that has animal-derived components, such as porcine and bovine-derived components, sponsors do need to keep in mind that the products that are manufactured using these animal-derived reagents should be tested so that to ensure that there is no transmission of adventitious agents from these animal-derived reagents.
Thank you so much. Patricia, what do sponsors need to do to perform xenotransplantation?
Xenotransplantation requires an investigational new drug application or an IND. The requirements for these studies are outlined in the guidance for early phase clinical trial design. However, before submitting an IND, we encourage sponsors to review the requirements for preclinical studies needed to support an IND and the guidance for xenotransplantation. I've provided links to these guidances on my slide 9 of my talk. We recognize the challenges that developing xeno products entail. Therefore, we recommend that you seek product-specific recommendations for specific indication by seeking early input from the FDA. I remind you that we offer interact and pre-IND meetings. Again, please refer to the links provided at the end of my talk for additional information. Also, since the animals are evaluated through CVM, we remind you that you need to have conversations with CVM as well. Thank you.
Thank you so much. Next, we have a question for Judy. Does the agency require that the pigs be knocked out for PERV?
At this point in time, there is no requirement for knocking out PERV. However, we do have recommendations based on the scientific literature that pigs that are express PERV AC recombinants are not used, because it's been reported in the literature that those pigs PERV is 500 times more effective in AC recombinants than in type PERV A only. So our current recommendation is to not include those animals. Thank you.
Sounds great. Next is a question for Archana. What is the reliability of the rabbit pyrogen test when it be requested as opposed to reliance on more current endotoxin standards?
Great question, Irina. So if using an alternative method to the rabbit pyrogen endotoxin test, to support the use of these alternative test methods, the sponsors need to provide data to demonstrate the method sensitivity, and they also need to validate the assay prior to the licensure. And as described in 21 CFR 610.9, the alternative methods must be shown to provide assurance of the safety and purity of the product equal to or greater than the assurance provided by the method specified.
Thank you so much. Next question is for Patricia. Is there a long-term follow-up protocol requirement similar to the gene transfer studies?
Yes, long-term follow-up will be required. It's addressed in the xenotransplantation guidance, and this will be, as most decisions, based on the specific product and intended use.
Thank you so much. Let's move back to Judy now. Is there any flexibility in the expectation for sampling, archiving, and the length of follow-up for patients receiving xenotransplantation products?
There is flexibility depending on the xenotransplant product. So for example, if your xenotransplantation product are human cells that have been exposed ex vivo to a well-characterized cell line, it's possible for you to have flexibility in those expectations stated in the PHS and FDA guidances on xenotransplantation. Where there is higher risk, such as the extracorporeal perfusion devices, then we would be a little bit more aggressive in our expectations because of the length of time that the human would be exposed to the animal cells. So there's a sliding scale, and the expectation is that you would discuss your plan with the FDA early on so that you can plan accordingly. Thank you.
Thank you so much. And this was a wonderful response to help us gain some clarity. The next question is for Archana. For a cell and device combination product, where should the device information be submitted in the BLA? And are there any FDA guidances for xenotransplantation device combination products?
Yes, sorry. Yes, Irina, there are guidances. So the Office of Combination Products and involved centers works on formatting certain recommendations for the licensure applications for these combination products to facilitate the review. And there is a 2022 guidance on this topic. It's called Guidance for Industry ECTD Technical Conformance Guide. Thank you.
Thanks so much, and all of these are available on our website, which is a wonderful resource. Patricia, can you please provide a list of current applications for xenotransplantation products?
So with every file, the submission of files is confidential under the FDA's rules. However, investigators and companies are required to list their IND applications under clinicaltrials.gov. So you can find those listed INDs within that website.
Thank you so much. The next question is for Judy. The audience participant is wondering if fecal transplants that are used to introduce bacteria to humans through the anal canal can be classified as xenotransplantation.
They are not qualified, considered to be xenotransplantation products because, you know, they're bacteria and not animals. And the Office of Vaccines at CBER is responsible for oversight of those products. Thank you.
Sounds great. Thank you. And it sounds like we have just a couple of minutes for maybe one more question for Archana. What are the CMC expectations for xenotransplantation products that are in encapsulated form? Is it the same as for xeno products that are not encapsulated?
That is correct. The same regulations apply to the encapsulated xenotransplantation product as they would for the non-encapsulated xeno products. And if the encapsulation portion of this product is a device, that device portion of this combination product will be reviewed as per the device regulations.
Sounds great. Thank you so much. It looks like we can all head out to lunch now until 1 o'clock. Thank you so much to our speakers for the wonderful presentations as well as the Q&As, and thank you to our audience members for your participation and wonderful questions. Enjoy your lunch.
Welcome back, everyone, to this CBER session that features 3 of CBER's staff members. Dr. Ann Rousey will present OTAT's Stakeholder Outreach and Patient Engagement Program. Dr. Manuel Osorio on the CBER Advanced Technologies team, and Ms. Crystal Melendez giving an overview of FDA-EMA Parallel Scientific Advice Program for Cell and Gene Therapy. Introducing them in the order of presentation is Dr. Ann Rousey, who is an Associate Director for Policy in OTAT. Dr. Rousey supports OTAT's stakeholder engagement efforts and coordinates its proactive patient outreach program. As part of OTAT's patient-focused outreach, Dr. Rousey has launched the Regenerative Medicine Therapy Outreach and Education Program, including the #RegenMedEd, patient webinar series, and annual patient workshops. Before moving to CBER, she was a science policy analyst in the Office of Therapeutic Biologics and Biosimilars in CDER, where she provided scientific guidance to ensure consistency and agency recommendations across biosimilar development programs. Dr. Rousey also has past experience spearheading communications, covering CDER's regulatory and research programs for the Office of Communications, OCOM. During her time in OCOM, Dr. Rousey wrote and directed manuscripts for high visibility, peer-reviewed journals, and led the OCOM editorial team. Next will be Dr. Manuel Osorio, who is currently the lead for CBER's Advanced Manufacturing Technologies. Prior to this role, Dr. Osorio was a researcher and reviewer in the Office of Vaccines Research and Review in CBER. He received a Bachelor's of Science degree from UCLA in biochemistry and received his PhD degree in cellular immunology from the University of California at Santa Cruz. He has also completed a postdoctoral fellowship at the National Institutes of Health, NIH. Last but not least, presenting is Crystal Melendez. Crystal is a regulatory project manager in the Division of Regulatory Management in OTAT. Crystal received her Bachelor's of Science degree in Medical and Research Technology at the University of Maryland School of Medicine and a Bachelor's of Science degree in Nursing from Stevenson University. Prior to joining the FDA, Crystal worked in the NIH in the Transfusion Medicine Department's HLA Immunogenetics Laboratory, specializing in bone marrow, cord blood, and stem cell transplantation. And even prior to that, she was at the University of Maryland Medical Center in Baltimore, Maryland, where she was a medical technologist in the Immunogenetics Laboratory, and is where she began her nursing career in the acute medicine unit. She continues as a practicing nurse at an acute physical rehabilitation hospital, specializing in stroke, traumatic brain injury, spinal cord injury, and amputation. It is my honor to introduce you all to Dr. Rousey, Dr. Osorio, and Ms. Melendez.
Hello, everyone. Thanks for hanging in there and joining us for the post-lunch session on the last day of the REdI workshop. My name is Ann Rousey, and I am an Associate Director for Policy within FDA's Center for Biologics Evaluation and Research in the Office of Tissues and Advanced Therapies, or OTAT, as I'm sure you've heard. I'm pleased to be speaking with you today about a very important topic, stakeholder engagement and patient outreach, and specifically how FDA is working to bring the patient voice to the forefront. So I'll start off my presentation today by providing a quick refresher on OTAT and regenerative medicine, and then I'll provide a brief overview of OTAT stakeholders and some of our upcoming outreach efforts. Then I'll focus in on patient engagement activities at the agency and provide specific examples of our work in OTAT to educate and engage patients and advocates in the regenerative medicine space. By the end of my talk, I hope you'll have a better understanding of the variety of stakeholders with which OTAT engages, the purpose and value of patient engagement for advancing emerging areas of medicine, including regenerative medicine. In addition, you should be able to list some of the initiatives OTAT is leading to engage with patients and hear their input. So although we're almost finished, with day 2 of the biologics track. I'll start off by quickly refreshing everyone's memory that OTAT sits within the Center for Biologics Evaluation and Research. OTAT is one of 3 program offices responsible for regulatory oversight of biological products within CBER. The other 2 being the Office of Blood Research and Review and the Office of Vaccines Research and Review. OTAT is led by Director Dr. Wilson Bryan and Deputy Director Dr. Rachel Anatol. And you may have seen OTAT's mission statement just yesterday in Dr. Bryan's presentation, but I'd like to take a second just to highlight the last sentence in which it says OTAT's regulatory decisions are data-driven, impartial, and compassionate. Since joining OTAT in 2018, I've been struck by the compassion that reviewers from all disciplines have for OTAT's patient stakeholders in particular. OTAT's reviewers want to learn from patients, and they strive to fold this knowledge into their regulatory work as they help to ensure that medical products are safe and effective. Part of OTAT's responsibility is to provide regulatory oversight for regenerative medicine therapies, and sometimes I'll refer to those as RMTs during my talk. RMTs were defined by law in the 21st Century Cures Act, which became effective in 2016. And RMTs include cell therapies, therapeutic tissue engineering products, human cell and tissue products, and combination products using any such therapies or products. And FDA further interprets the definition of RMTs to also include human gene therapy products, including genetically modified cells that lead to sustained effects on cells or tissues. And also further interprets the definition to include certain xenogenic cell products. Definition of RMTs does not include certain human cell and tissue-based products that are regulated solely under Section 361 of the Public Health Service Act and 21 CFR Part 1271. For more information about these products, which we often call 361 products for short, I encourage you to view the recorded presentation on the HCTP regulatory framework from my colleagues Scott Brubaker and Safa Carendish from the REdI 2021 conference. Also excluded from the definition of RMTs are microorganisms like viruses, bacteria, and fungi that are not genetically modified. So, as you can tell by just the definition of these products, regenerative medicine is complex, and it's important to note that while regenerative medicine has been around for decades, it continues to evolve and progress through scientific advancements. Moving a little into FDA's role in regulating RMT products, as with small molecule drugs and other biological products regulated by regulated by FDA, OTAT rigorously reviews RMT products to ensure that they are effective and safe for patients throughout the product lifecycle, from development through approval and in the post-marketing setting. During development, OTAT provides oversight of clinical trials to help ensure people participating in the trials are protected. OTAT also helps advance the state of the science for RMT products by providing advice and education to product developers. And that's not just our industrial product manufacturing partners, but also researchers who are developing products in academia as well. Finally, stakeholder and patient engagement is a critical aspect of our work, and one in which I'll talk about in greater depth in the coming slides. But first, I'd just like to spend just a few slides giving an overview of OTAT's stakeholders and some of our efforts to engage with these groups. And our primary stakeholders fall into 3 categories: academic groups, trade organizations, and public-private partnerships, and patients and their advocacy organizations. So starting off first with these academic stakeholders, OTAT works with a variety of academic stakeholders, such as the National Center for Advancing Translational Sciences, or you've heard that group referred to as NCATS, and the Health and Environmental Sciences Institute, or HESI. OTAT works to help organize external workshops and provide regulatory perspectives at conferences and other meetings that are organized by these groups. Often OTAT staff members also participate in working groups and on steering committees such as the National Academies of Science, Engineering, and Medicine's Forum on Regenerative Medicine. And their role is to help shape the discussion on emerging scientific topics, and particularly in this case, those related to regenerative medicine. There are a fair few trade organizations devoted to advancing the field of regenerative medicine, and too many for me to mention in my talk today, but OTAT meets annually with these organizations to hear their positions on higher priority topics. And recently, OTAT conducted listening sessions with product sponsors and trade organizations to hear their ideas for us to work together to help expedite advances in cell and gene therapy. OTAT staff members are also providing regulatory perspectives via their participation in public-private partnerships like the Bespoke Gene Therapy Consortium, which launched late last year. As I mentioned in the previous slide, OTAT conducted some listening sessions with sponsors and trade organizations last year, and as part of this work, we identified two major goals to help expedite advances in cell and gene therapy, those being to, one, improve communications with all of our stakeholders, and also to increase capacity and efficiency in OTAT operations. So as some first steps toward addressing these goals, OTAT is updating and consolidating our web-based resources related to regenerative medicine therapies, and also updating content on OTAT's Learn website to support productive engagement with sponsors. We're also still collaborating with our trade organization partners to identify and facilitate areas or high priority topics that we would benefit from mutual learning on. And finally, just a teaser for what's coming up in fall. You can expect to see at least 2 public meetings from OTAT on gene therapies coming up in the fall, one being methods and approaches for capturing post-approval safety and efficacy data, and the other being patient perspectives on gene therapy products. So stay tuned for some of those outreach efforts coming up. So now that I've touched on our academic and industry stakeholders, I'm going to spend a few minutes diving into the work OTAT has been doing to engage directly with patients, caregivers, and advocates when it comes to regenerative medicine. So let's start by defining exactly what we mean when we say patient engagement. And it's pretty straightforward. Patient engagement means activities that the FDA and patients, their caregivers, and advocates take part in together. We at FDA want to hear about patients' personal experiences and their perspectives on living with a particular disease or condition, as well as patient needs and priorities when it comes to treatments. The patient voice is critically important to FDA, whether it's about regenerative medicine therapies or any other type of medical product, because it helps to inform FDA's mission of ensuring the safety and effectiveness of medical products. And I'll go into why and how FDA uses this information in the evaluation of medical products in the next few slides. But I'll just sum up by saying that the overall goal of the agency's patient engagement activities is to learn directly from patients. So to go a little deeper into the why does patient engagement matter to FDA? Well, to start, patients are at the heart of FDA's work, and that's true regardless of the disease or patient population in question. Further, patients are experts in what it's like to live with their disease or condition, and what it's like to use available treatments if there are any. For instance, they can tell us what symptoms have the greatest impact on their daily lives, the pros and cons of existing treatments, and what their quote-unquote ideal treatment would be. Patients can also best identify and articulate what is most important to them when it comes to treatments. What is a meaningful benefit to a patient? Is it fewer hospital stays? Is it fewer missed days of work or school? Is it being able to perform basic daily tasks independently? This is always going to depend on the patient population and their specific needs. Hearing directly from patients can help ensure we're understanding their needs so that we may facilitate development of products that address their chief complaints, and also so we can help to support design of clinical trials that factor in patient and caregiver needs and preferences. So finally, the how. OTAT and FDA more broadly has sought to standardize how we receive patient input. And I'll walk us through a few examples of some of the ways that we seek patient input and where patients can volunteer to engage with us. So starting off with the first item on the list here, advisory committee meetings. And these are often open to the public to discuss biologics license applications, or BLAs. You'll sometimes hear us say BLAs. During an AC meeting, a group of experts are asked questions about the BLA. They listen to information, and then they make a recommendation to the agency. And patients are included on these committees as special government employees, or SGEs, and they are voting members on the committee, and they consider the information as well as everyone else on the committee. Special government employees, or SGEs, can also provide input at the request of FDA review staff. And this can be patients, this can be caregivers as well. And patient or caregiver SGEs may be asked, for example, to provide their perspective on a certain aspect of drug development, such as how meaningful a proposed endpoint may be to them, or in the case of caregivers, to whoever they care for, or for patients, if they can speak directly for themselves, how meaningful that proposed endpoint would be to them in their daily lives. Another way we engage with patients is through public meetings and workshops. Patients and representatives from organizations, advocacy organizations, are often speakers and panelists at FDA meetings. And for a homegrown example, OTAT recently hosted a workshop, and it was just last month, where we had a panel discussion with advocates and patients to discuss their experiences participating in natural history studies. Patient-focused drug development meetings are another more formal way to receive patient input. PFDDs, as you'll hear us, we love acronyms in government, you'll hear us say PFDDs. These meetings are designed to get patient perspectives on their most significant symptoms and the impact of their condition on their lives, as well as to get their views on current approaches to treatments. And these days are the PFDD meetings are usually about a half day, and they're primarily composed of patients and caregivers. And there usually is live polling to get the views of all the attendees. And it really provides great insights into the view of a broad spectrum of patients who are living with the same or similar diseases and conditions. FDA also has meetings with patient organizations who are looking for specific input on work products like surveys or natural history studies or other methods that they're employing to conduct research to move the state of the science ahead for their particular disease or condition. And finally, the last thing you'll see on the list here is that FDA has been hosting listening sessions in collaboration with the National Organization for Rare Disorders, or NORD. FDA and NORD have hosted almost 30 listening sessions since 2018, and they're focusing on patients with rare diseases. And one final way that FDA and OTAT receives patient input that isn't explicitly stated on the slide is that patients and advocates can join in regulatory meetings with product sponsors. This is at the invitation of the product sponsor, but OTAT generally encourages sponsors to do this, to invite patient representatives and advocacy groups to attend their regulatory meetings with the agency. And on the same note, OTAT is encouraging patients and their advocates to approach sponsors and request to attend these meetings. And not only does this interaction give us in OTAT an opportunity to hear the patient voice on specific issues, it also helps patients to get a clearer view of our processes and potentially of any concerns we may have. OTAT believes that we need to increase our outreach to patients caregivers, and advocates so that they may better understand the regulatory process, and hopefully this will lead to greater confidence in approved products. So for the last few minutes of the presentation, I'm going to talk about one such program underway within OTAT to reach out to the patient community. And this is our Regenerative Medicine Therapies Outreach and Education Campaign. The goals of this campaign are to, first and foremost, broaden public understanding of regenerative medicine therapies. It's to help us understand patient beliefs and perceptions about regenerative medicine therapies, as well as their information needs and communication preferences. In the lower left-hand corner, you can see another goal is for us to educate patients, caregivers, and advocates about regenerative medicine therapies. And this is really so they can help make better healthcare decisions for themselves and their loved ones. And finally, an overarching goal of a lot of our work, and this in particular, is to enhance interactions between OTAT and patients. And one way OTAT is seeking to achieve these goals is by hosting public workshops and meetings, like our annual patient engagement workshop. You heard me mention that a little bit earlier, but these workshops are half-day virtual events. We hold them in spring. We had our first in 2021, and we recently hosted the second annual workshop last month. And you can find recordings of these workshops online. The goal is to bring together hundreds of patients, caregivers, advocates, and other key stakeholders to discuss ways that we can work together to advance regenerative medicine. This year, our workshop focused on natural history studies and why these studies are so critical for advancing regenerative medicine therapies like gene and cell therapies. Another public event OTAT hosts is our quarterly Regen Med Ed webinar. And Regen Med Ed stands for Regenerative Medicine Education. In a Regen Med Ed webinar, you can expect an hour-long deep dive into a specific topic related to regenerative medicine. And we often feature patients and caregivers to tell their stories. For example, just this past March, we hosted a Regen Med Ed webinar, and this was on the critical role of patients in advancing gene therapy treatments for rare diseases. We heard from a patient with hemophilia who participated in a gene therapy clinical trial, as well as an advocate from NORD. Finally, I'd just like to talk a little bit more about our meetings with patient organizations. And these often occur because the patient group has a particular project on which they'd like the agency's comment, or a work product where they would like to share the results directly with the agency. And so some examples of these products are things like meeting reports, such as the Voice of the Patient reports that summarize externally led patient-focused drug development meetings, or reports that spotlight scientific or policy meetings held by advocacy organizations. Another work product that patient advocates often share with us are their patient survey results. For example, we recently met with a group who provided their plan for surveying patients on their preferences for participating in clinical trials for gene therapy. So it's not just, once they have the results, and they share those with us, but they also will meet with us in advance to ask questions about their study design to be sure that the information that comes out of it could be useful to the agency. Another work product that advocacy organizations like to share with the agency are white papers or journal articles, and this is usually following the study. There's usually a summary report that shows the methodology and the results they have received from their surveys and other patient studies. Moving down the list, natural history study reports, and also protocols and development. It's similar to what I was just saying about the patient surveys. OTAT recently met with a patient group who had questions about the utility of their observational study design and the potential for data to support future gene therapy clinical trials in that space, and what were some of the potential outcomes that they could expect from a regulatory perspective from that work. Finally, the last thing you'll see here on the list is that patient organizations could also draft a guidance document related to patient experience related to their disease or condition of interest and send that to us as well. And just a note, to speak from my experience in facilitating these types of meetings for OTAT, even though our reviewers experience a very heavy workload, they make time and prioritize these interactions with patient organizations. Sometimes it might take some time for us to get these meetings on the calendar, but they are important to reviewers, and you have their attention when these meetings take place. So, just to wrap things up, I just want to share a few resources where you can find a little more information about OTAT's patient education and stakeholder outreach campaign. You can follow CBER on Twitter to stay informed about upcoming events and meetings. You can find our past webinars and workshops linked on FDA's website. And you can also find more information about regenerative medicine on the cell and gene therapy web pages of FDA's website. So to summarize, just like to point out 3 things. The first being that stakeholder engagement is critical because it helps inform OTAT's mission and FDA's public health mission as a whole. We talked about many different opportunities for patients to engage with the FDA, and I want to highlight that it's also about finding the right channel so patients, advocates, and the agency can gain the most from all of our interactions. And lastly, when it comes to novel therapies, like gene and cell therapies, where the state of the science is still rapidly evolving, it's so important for FDA to understand patient perspectives. Okay, so moving on to our first challenge question. Which of the options below would not be considered a relevant type of patient input for the FDA? A, patient perspectives on the most burdensome or difficult symptoms of a disease. B, patient perspectives on risk tolerance of a potential treatment. C, patient perspectives on the risks of participating in a clinical trial. Or D, patient perspectives on drug pricing. So hopefully this one won't be too difficult because I didn't talk at all about drug pricing, but the answer is D, patient perspectives on drug pricing. Drug pricing is out of FDA's purview. Moving to challenge question number 2, true or false? OTAT's Regen Med Ed webinar series is held annually. I'll give you a second here before moving on to the answer, and that is false. It's a bit of a trick question. OTAT's Regen Med Ed webinars are held quarterly. Our patient workshops are held annually in spring. So thank you all for your attention. Here's my contact information. If you have any questions, please reach out. And I look forward to the panel discussion. Thanks so much.
Thank you, Jen, and thanks to the organizers for the invitation to speak to you about a relatively new initiative in the Center for Biologics Evaluation and Research, which is a CBER Advanced Technologies team. This initiative is part of a larger program to promote the development and adoption of advanced manufacturing technologies for CBER-regulated products. My name is Manuel Osorio, and I am the lead for the CBER Advanced Technologies team. I am in the immediate office of the director in CBER. After this presentation, I hope you will, if not already, understand the importance of advanced manufacturing to public health. It is my hope that you can learn and appreciate what CBER is doing through various activities to promote the development and adoption of advanced manufacturing technologies for pharmaceuticals, and that you will become familiar with and explain the purpose and scope of interactions with the Advanced Technologies team. So let's start by defining advanced manufacturing. Advanced manufacturing is an innovative pharmaceutical manufacturing technology or approach which has the potential to enhance drug quality, improve the reliability and robustness of the manufacturing process and supply chain, and increase timely access to quality medicines. It can integrate novel technological approaches. It can use established techniques in a new or innovative way, or apply production methods in a new domain where there are no defined best practices or experience. Advanced manufacturing is applicable to chemical as well as biological entities. Advanced manufacturing provides benefits for industry as well as consumers. It has the potential to enhance product quality as a result of a reliable and robust manufacturing process. It can reduce manufacturing costs due to factors such as smaller manufacturing footprint. The manufacturing time and time to market post-approval can be reduced by significantly reducing the need for scale-up. It can prevent shortages of needed medicines resulting from improved product quality. It can improve emergency preparedness because it allows for agility and flexibility to manufacturing that can quickly respond to demand. This slide shows the product classes that are regulated by CBER. The Center's experience with advanced manufacturing technologies has been limited. However, we know there is strong interest in implementing advanced manufacturing technologies for advanced therapies, such as cell and gene therapies. But we're also starting to see interest in using advanced manufacturing for more traditional product classes, such as vaccines and blood-derived products. Before I go into the details of the CBER Advanced Technologies team, I want to provide a wider perspective on the different initiatives under the CBER Advanced Technologies program, with the goal of promoting the development and adoption of advanced technologies to modernize pharmaceutical manufacturing. The program was established in June of 2019, and it consists of 3 major initiatives. The first initiative under this program is funding of advanced extramural research and development projects to support regulatory science and innovation. Of course, the Center also supports intramural research on advanced manufacturing. However, those projects are not funded through this program. The extramural projects that we fund address knowledge and experience gaps, identify for emerging manufacturing and testing technologies, and support the development and adoption of such technologies in the biological product sector. Since 2018, CBER has awarded several grants and contracts to support research projects to study improvements for advanced manufacturing biological products. I believe we have committed over $25 million for this purpose so far. Some of the projects we support are developing technologies such as novel manufacturing approaches, for example, critical quality attribute discovery, advances in purification processes, and continuous production of cell therapy products. We're also funding projects on end-to-end manufacturing of gene therapy products, so this will be continuous manufacturing of AAV vectors. Also, we are supporting projects on continuous manufacturing of vaccines, 3D bioprinting for tissue engineering, and process modeling and simulation. We are also working on building the scientific and regulatory expertise necessary to evaluate these emerging technologies. For now, we're doing this through training seminars on topics related to advanced manufacturing. We usually invite our external grantees for presentations to our CBER staff as a training exercise. Now, I should note here that there are also ongoing efforts within the review offices in CBER to develop training material on advanced manufacturing topics focused on the product classes that are reviewed by the particular office. Towards the end of my presentation, I will also describe additional activities at the agency level as well as internationally aimed at building the appropriate regulatory framework to evaluate advanced manufacturing technologies. The third initiative, which is the focus of today's presentation, is the CBER Advanced Technologies Team, or CAT. CAT was established in June of 2019 to promote dialogue, education, and input among CBER staff, and for that matter, FDA staff, and between CBER and prospective developers of advanced manufacturing technologies. It provides an interactive mechanism for developers of advanced manufacturing and testing technologies to discuss with CBER issues related to the implementation of these technologies in the development of CBER-regulated products. These interactions generally occur prior to regulatory submission. CAT interactions for discussing innovative approaches to biologic product development, such as novel technologies, including manufacturing and control strategies that can have a significant impact on product development and with potential regulatory implications. This includes manufacturing and analytical methods for those products or classes of products for which the center has limited experience with the manufacturing or development process. CAT may also consider other general topics such as innovative facility design and innovative clinical trial design. It is important to understand that CAT interactions are not for discussing product-specific, highly technical aspects. As you will see in the next slide, this type of discussions are better suited for other types of meetings. You may already be familiar with the various interactions you can have with the FDA throughout the product development pathway, from the pre-IND stage to BLA submission. And of course, interactions can occur post-approval. For the development of novel therapies and innovative approaches to product development, we highly encourage early interactions with the agency to address any potential regulatory issues at an early stage of development. CBER has two types of informal interactions that can occur early in development, and we highly encourage you to take advantage of these. One is the Interact meetings, which I'm not going to describe in detail during this presentation, except to say that these meetings are for the purpose of discussing product-specific issues related to CMC, pharmacology, toxicology, and clinical study design for first-in-human use. A key requirement for Interact meetings is that a product needs to have already been identified prior to submitting a request to the center. However, if you are a technology developer or innovator with a technology that could significantly impact manufacturing of a product class or multiple types of products, but have not yet identified a specific product to develop, then the only option to obtain feedback from CBER would be through a CAT interaction. CAT fills that gap where you can obtain regulatory feedback on the implementation of an advanced technology for manufacturing CBER-regulated products, even before a specific candidate has been defined. CAT meetings are well suited for discussing platform technologies for manufacturing biologics. Now, I just want to point out a couple of important pieces of information that are related to this information on the slide. One is that both CAT and Interact meetings are not formal, and therefore any advice given through these interactions is non-binding for CBER. Also, CAT meetings are not required for soliciting an Interact meeting or a pre-IND meeting or an IND meeting. So who is part of the CAT team? The team consists of a small cross-functional group representing CBER leadership, relevant policy review and inspection programs. Its members come from various offices within the center, such as the Office of the Director, the Office of Compliance and Biologics Quality, Office of Tissues and Advanced Therapies, Office of Vaccines Research and Review, and Office of Blood Research and Review. Not listed here is the Office of Biostatistics and Pharmacovigilance. However, if we received a CAT request that requires an input, we can easily reach out to them for their feedback. As CAT interactions are relatively new, I think it would be useful to take a few minutes to go over the process for submitting the request, the information that should be included, and how requests are evaluated, and finally what you might expect to get back from CAT. To submit a request, please visit the web portal, which you can find through the link shown on the slide. Alternatively, you can just do a web search for CBER Advanced Technologies Team, and you will get to the portal. So what information do you need to include with your request? You need a two-page backgrounder that includes figures and tables that provides the following information: a brief description of the technology or product class, a brief explanation of why the technology or product class is substantially novel and unique, a description of the impact of the technology or product class in terms of improved biologic product manufacturing, characterization, quality, safety, or efficacy, a summary of the manufacturing development plan, and this is very important, any questions regarding perceived regulatory or other challenges for implementation. Once the request is received, it will go through an initial evaluation by the CAT coordinators to determine whether the request is within the scope of CAT meetings. Then we will decide which office or offices within the center will be the lead offices in providing feedback. We also discuss submissions during regular internal CAT meetings, where we can obtain additional feedback from the multidisciplinary team. Following review of the request by our subject matter experts, there are three possible outcomes. One is that a CAT meeting is granted. A big part of the decision to grant the meeting is the degree of familiarity the center has with the specific technology. In addition to providing informal feedback to the technology developers or prospective sponsors, CAT meetings are a unique opportunity for CBER staff to learn about innovative approaches to product development that we may see in a future IND submission. Once the meeting is granted, we try to schedule it within 3 weeks of notifying the requester of our decision. For the meeting, the requester has the option to do a presentation to provide additional details about the technology and to drive the discussion during the meeting as well. The slide deck should be sent to the CAT coordinator at least 3 working days in advance of the meetings to give our subject matter experts time to review the additional material. If a meeting is not granted, we will provide written feedback to the questions submitted with the request. This is why it's important to include questions with the background material when submitting the request. If the request is for product-specific questions or the questions require highly technical discussions, then we will recommend that the requester seek a different type of meeting, such as an interact meeting, pre-IND meeting, or IND meeting, which are all product-specific discussions with. Some of the technologies we have discussed to CAT interactions include the following: continuous manufacturing for vaccines, AAV vectors, exosomes, fully closed, automated, scalable, and remote control systems for manufacturing cell therapy products, innovative tools for assessing vaccine immune responses, improved cell lines for vaccine antigen production and AAV vector manufacturing, use of artificial intelligence and advanced imaging technologies for real-time product quality assessment, multi-product manufacturing facility design, and continuous processing of blood-derived products. So what has been our experience with CAT thus far? As I mentioned earlier, CAT is relatively new, and it is a program that has been received with enthusiasm by stakeholders, but we have had some struggles. There has been a lot of interest from the technology developers and manufacturers in using this mechanism to establish a dialogue with CBER to discuss the implementation of these technologies. In the last 3 years since CAT was established, we received over 60 CAT requests and have granted 11 meetings. Before the pandemic, meetings were face-to-face. Lately, we've been having them virtually, but we do hope to get back to face-to-face meetings in the near future. For requests that were not granted a meeting, we provide a written feedback via email, as I explained earlier. There have been some pains, mostly due to the pandemics, as FDA has had to redirect resources to deal with the pandemic. And this had a significant impact on the availability of CBER subject matter experts to address the CAT requests that we received. When the program first started, our goal was to provide a response to the request within 6 weeks of receipt. Unfortunately, lately that has been rather difficult to achieve. However, the future looks bright as the availability of our subject matter experts to address CAT requests slowly improves, and we're actually already addressing some CAT requests within a short time following receipt. In addition to providing a mechanism for technology developers to engage CBER to discuss regulatory and technical issues related to the implementation of advanced manufacturing for CBER-regulated products, the Center, through CAT, is collaborating at the agency level as well as internationally to facilitate the adoption of advanced manufacturing technologies. In June of 2021, CDER and CBER established the Center for the Advancement of Manufacturing Pharmaceuticals and Biopharmaceuticals, or CAMP-B. The mission of this program is to accelerate the development, implementation, and evaluation of advanced manufacturing by establishing science and risk-based standards and policies, and to advance drug development science, and also to train a world-leading regulatory workforce through strategic partnership, engagement, and communication. This diagram represents the 6 areas where we see potential collaboration between the centers. And it's also important to note that this is not a collaboration just between the Center for Biologics and the Center for Drugs, for we expect other groups within the FDA, such as the Office of Regulatory Affairs, as well as other centers to participate in this program. The 6 areas where we see collaboration are shown on the diagram, but I just want to highlight a couple of them. For pre-submission engagement, CDER has the Emerging Technology team, and CBER has the Advanced Technologies team. And we're beginning to collaborate informally on engagement with prospective sponsors, implementing technologies that could have applicability for the manufacture of both CDER and CBER regulated products. Another area where we are collaborating is on developing a regulatory framework for evaluating advanced manufacturing technologies. Recently, CDER announced a new initiative called FRAME, which stands for Framework for Regulatory Advanced Manufacturing Evaluation, to develop the appropriate regulatory framework to evaluate emerging technologies such as distributed manufacturing and artificial intelligence. CBER is also actively involved in this initiative to align with CDER on building this framework to evaluate these emerging technologies. Through the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, or ICH, CDER and CBER, along with representatives from other regulatory agencies and industry groups, are working to draft the ICH Q13 guidance on continuous manufacturing of drug substances and drug products. The objective of this effort is to provide a harmonized guidance for the development, implementation, and assessment of continuous manufacturing technologies used in the manufacture of drug substances and drug products. This guidance will apply to continuous manufacturing of chemical entities and therapeutic proteins and the conversion of batch manufacturing to CM for existing products. Although the focus is on chemical entities and therapeutic proteins, which are products regulated by CDER, the guidance is forward-looking, and the principles outlined in the guidance may also apply to other biological and biotechnological entities, such as those that are regulated by the Center for Biologics. And just to summarize, I hope that I convinced you that advanced manufacturing can improve pharmaceutical manufacturing and ensure the availability of good quality medicines and play an important role in public health. Also, that CBER is committed to supporting the development and adoption of advanced manufacturing technologies for CBER-regulated products, and we do this through various activities such as extramural funding and CAT interactions. CBER is collaborating internally and internationally to build the scientific expertise and regulatory framework that will be required to evaluate emerging technology. Specific examples of this are the CAP-B program as well as the ICH Q13 technical document. We will now do a check of your knowledge with 2 challenge questions based on the information I provided in my presentation. The first challenge question is the following. Which of the following statements is not true? A. CBER discourages the implementation of advanced manufacturing technologies for biologics because there is no regulatory framework for their evaluation. B. CBER encourages the development and adoption of advanced manufacturing technologies for biologics. C. Advanced manufacturing technologies can improve the quality and availability of needed medicines. D. Advanced manufacturing technologies can improve emergency preparedness. I will give you a few seconds to choose the correct answer. And the correct answer is A, which is not true. Quite the opposite, actually. CBER actually encourages the development and adoption of advanced manufacturing technologies for biologics. As I explained during my presentation, we do this through various activities to promote the development and adoption of these technologies. The second challenge question is the following. Which of the following statements about CAT interactions is true? A. CAT meetings are formal meetings and therefore any advice provided is binding. B. CAT meetings are for discussing product-specific regulatory issues related to CMC, toxicology, and clinical study design. C. CAT meetings are required before filing an IND submission. D. CAT meetings are informal and therefore any advice provided is non-binding. I'll give you a few seconds again to choose the correct answer. And the correct answer is D. CAT meetings are informal meetings and therefore any advice provided is non-binding for the agency. This concludes my presentation. I thank you for your attention. We will have a Q&A at the end of the session, and I will be happy to answer your questions. You can also reach out to me directly with any other questions you may have at the email address that's shown on the slide. The link at the bottom of the slide is to the CBER Advanced Technologies Program, and the website will provide additional links to the CAT portal for submitting CAT requests, as well as information on the extramural projects that we are currently supporting. Thank you once again.
Thank you so much for that great introduction. I'm pleased to give this two birds with one stone approach for seeking scientific advice. In this presentation, I will be giving an overview of the FDA-EMA Parallel Scientific Advice Program for the Center for Biologics Evaluation and Research. The purpose of the Parallel Scientific Advice Program is to provide a concurrent review and exchange of advice between two regulatory agencies, the U.S. Food and Drug Administration, or FDA, and the European Medicines Agency, or EMA. The advice given to sponsors in this program focuses on key scientific issues that may come up during the development phase of new medicinal products. These interactions are expected to increase dialogue between FDA, EMA, and sponsors early in the life cycle of a new product to develop an understanding of regulatory decisions and to optimize product development. The learning objectives for this presentation include discussing the FDA-EMA PSA agreement, identifying the best candidates and eligible products, identifying the benefits to PSA interactions and meetings, learning how to submit a PSA request, identifying what information is submitted in a PSA request, reviewing the estimated timeline for PSAs, and understanding the process as well as how PSA meeting requests are managed in CBER. So let's begin with a quick history lesson. On September 17, 2004, the FDA and EMA agreed to launch a pilot program to provide parallel scientific advice, or PSA, and establish the policies and procedures for the program. The pilot program started on January 1, 2005, and within a year saw a significant increase in PSA activities. This increase led to an indefinite extension of the PSA program the following year in 2006. Finally, in 2020, there was a revision to CBER's SOP-P 8001.6 to incorporate new procedures, which I'll discuss in today's presentation. There are some PSA basic guidelines to follow. The PSA process is voluntary, and the request may be initiated by the sponsor, FDA, or EMA in cooperation with the sponsor. It's intended to address a single set of questions submitted by a sponsor for which the sponsor desires to have scientific input from both FDA and EMA. It should not be viewed as an ongoing series of consultations. PSA should focus on specific questions or issues involving the development of an eligible product. Continuing with some basic guidelines, PSAs do not fall under the Prescription Drug User Fee Act, referred to as PDUFA, and are not subject to the performance goals for scheduling and holding PDUFA meetings. However, they are similar and align closely to a CBER pre-IND Type B meeting and corresponds to the 70-day timeline of EMA Scientific Advice Working Party, or SAWP. Next, we'll review some key factors that determine the best PSA candidates. For FDA, the PSA aligns closely to sponsors in pre-IND or end of phase 2, depending on the stage of product development. The product should be considered an important medicinal product such as unmet medical needs, and the indication should lack development guidelines or have significantly different guidelines for FDA versus EMA. The candidate may be developing biosimilars or products with significant clinical safety, animal toxicology, or unique manufacturing concerns that could impede further product development. And lastly, it should treat rare diseases and special populations. Now we will move on to discuss the FDA-EMA agreement for PSAs. The PSA takes place under the provision of the confidentiality arrangement between the FDA and EMA with the sponsor's authorization. Both agencies remain committed to meeting domestic process and review goals and timeframes and exhibit as much flexibility as possible in scheduling PSA meetings. This helps to avoid adversely affecting or impacting either agency's ability to meet its formal domestic performance expectations. The following are CBER products eligible for the Parallel Scientific Advice Program: cell therapy products, gene therapy products, tissue engineered products, vaccines, and plasma derivative products. You may be wondering why a sponsor would request a PSA meeting. There are several benefits for requesting parallel scientific advice. PSAs allow for an increased dialogue and discussion between both agencies and the sponsor at various points in a new product's life cycle. It gives the sponsor a better understanding of the foundations of the advice given, as well as the opportunity to truly advance and optimize their product's development. It helps reduce and avoid unnecessary testing replication or divergence in testing methodologies. And sponsors save time and resources by submitting a single meeting request to both agencies. Sponsors wishing to nominate a product for parallel scientific advice should send one single request for PSA letter to both emails listed here. In the PSA letter request, the sponsor should provide the following information: the product and development, an explanation of why a discussion with the agencies would be beneficial to the product's development, a list of specific questions requiring clarification from the agencies, the desired goals for the meeting, and an explicit authorization for the agency's comprehensive exchange of all information relevant to the product, including trade secret information. There are a few considerations to keep in mind regarding PSA requests. A PSA request does not guarantee the PSA procedure will be granted. One or both agencies may decline to participate. And If a sponsor's request for PSA is not granted, the sponsor is free to pursue a scientific advice procedure with each agency individually, following each agency's normal procedural rules. Because a PSA request involves so many moving parts, there are several parties that must be involved. We'll briefly go over their roles here. The Senior Advisor for International Affairs, SAIA, for FDA oversees the PSA procedure within CBER and works closely with the Foreign Regulatory Communications Coordinator, as well as the Product Office Lead or Regulatory Project Manager. The Foreign Regulatory Communications Coordinator, or FRCC, for FDA, executes the PSA procedure and works in conjunction with the Senior Advisor for International Affairs, EMA, and Regulatory Project Manager to coordinate the request and scheduling of meetings. This role also secures sponsor authorization to permit a comprehensive interagency exchange of all information relevant to the subject product. The Product Office Lead, or Regulatory Project Manager, is the point of contact for sponsors and facilitates the CBER PSA meetings with the Senior Advisor for International Affairs, the Foreign Regulatory Communications Coordinator, and EMA. The FDA CBER review team includes product or CMC reviewers, pharmacology toxicology reviewers, clinical reviewers, and consults as needed. There is also an EMA point of contact and assessors or Scientific Advice Working Party, also known as SAWP. The sponsor and 2 patient representatives may participate in the trilateral meeting and post-trilateral meeting, briefing if the sponsor and FDA agencies both agree. Again, the Parallel Scientific Advice Program does not follow any of the Drug User Fee Acts, so the PSA timelines shown here are target dates. Both agencies will work together closely and remain committed to meeting the review goals and timeframes by being flexible and scheduling meetings without affecting performance expectations. As seen here, the sponsor may submit the request at any time and will work closely with the Senior Advisor for International Affairs for FDA and the point of contact for EMA to determine the best time frame to submit their meeting package. Once the meeting package is submitted, the trilateral meeting with the sponsor will occur approximately day 60. The PSA process officially begins when a single PSA request letter is sent to both agencies and is reviewed for appropriateness by each respective agency. As mentioned previously, the PSA process aligns with CBER's Type B meeting timeline and EMA's Scientific Advice Working Party procedure, but there is some flexibility with scheduling of the meetings and the meeting package submission timeframe. CBER's notification to grant or deny a PSA request is usually made within 14 days, but it could take up to 21 days. The notification by EMA will be made within 10 days of the request. If both agencies grant the PSA request, the sponsor will receive a teleconference confirmation letter for the meeting from both CBER and EMA. CBER's confirmation letter will include the trilateral meeting date and time with the sponsor as well as the date agreed for the receipt of the meeting package. Once the meeting package is submitted and received, the PSA review begins with each agency separately. The day the meeting package is received is considered day 0. Internal meetings or discussions occur separately for both agencies approximately 15 to 30 days after receiving the meeting package. The bilateral meeting with both agencies will occur about 10 to 14 days after the internal meeting. Preliminary feedback from the FDA and a list of issues from EMA are then sent to the sponsor separately. which is approximately 2 to 7 days before the trilateral meeting. The 90-minute trilateral meeting is held on approximately day 60 from the meeting package receipt date. Due to time differences, meetings with EMA must start between 8 a.m. and 12 p.m. Eastern Standard Time. The meeting attendees for trilateral meetings include FDA, EMA, the sponsor, and, if requested and approved by all parties, 2 patient representatives. Following the trilateral meeting, FDA and EMA will have a 30-minute debriefing discussion. Within 10 days of the trilateral meeting, EMA will provide the sponsor a final advice letter, and FDA will issue the final meeting summary by day 30. Lastly, final PSA advice letters will be exchanged between agencies. Each agency will provide independent advice on the questions posed by the sponsor. The advice of each agency may be different after the joint discussion. Sponsors should not expect to receive similar recommendations from the 2 agencies regarding drug development issues, nor should they expect to receive similar agency decisions regarding marketing applications that have undergone PSA. Each agency will retain its individual regulatory decision-making authority regarding drug development issues and marketing applications. However, both agencies will strive to provide PSA responses that are convergent. There are some important best practices a sponsor should consider when requesting a PSA. A PSA should not be the first encounter with FDA. A sponsor should have a prior interact, pre-IND, or IND with FDA. This will ensure that the PSA focuses on global development. Sponsors should be understanding and flexible of the timelines, review goals, and time differences for both agencies operating across 2 continents. Sponsors should ensure the appropriateness of a PSA request to make sure their product is eligible and if they are a good candidate. A single PSA request should be submitted separately to both agencies. Meeting packages should be complete and clear on which questions are for FDA, EMA, or both. And sponsors should be well prepared for the trilateral meeting using the preliminary feedback and prioritizing the questions to foster effective discussion while keeping in mind the 90-minute time frame. To summarize, the following PSA process, sponsors should have a clear understanding of FDA and EMA's respective requirements and perspectives regarding the development program and, if divergent, the reasons for the divergence. Scientific advice given during the PSA process to sponsors promotes global product development. PSAs are not intended to provide combined or joint advice from the 2 regulatory agencies. There is an opportunity for increased dialogue and possible convergence in terms of development requirements between agencies, and each agency will provide their independent advice to the sponsor separately. Now I would like to ask the audience 2 challenge questions. Challenge question number 1. The PSA trilateral meeting occurs approximately how many days after receiving the meeting package? A, 14 days. B, 30 days. C, 60 days. D, 90 days. Take a second to consider which answer you think is correct. I hope you selected C, 60 days. The second challenge question. Which of the following statements is not true? A, the PSA process is voluntary. B, PSA is viewed as an ongoing series of consultations. C, PSA is initiated at the sponsor's request. D, PSA is intended to address a single set of questions. E, PSA can be initiated by EMA or FDA in cooperation with the sponsor. Take a second to consider which answer you think is correct. I hope you selected B. PSA is viewed as an ongoing series of consultations. On this resources page, you will find guidance documents and links for PSA policies and procedures, as well as the emails for FDA and EMA for sending your PSA request letter. I would like to close out this presentation by encouraging eligible sponsors seeking global drug development to submit a request for parallel scientific advice. That concludes my presentation. Now we will move on to the Q&A section.
Amazing. I want to take this moment to thank you, Anne, Manuel, and Crystal for your wonderful presentations. Now, if you wouldn't mind, Anne, would you start us off in our Q&A with the following question we just received? And how can a patient or advocate submit topic ideas for Regen Med Ed webinar or workshop?
Hi, Jen. Thanks for that question. It's amazing. I'm really happy that folks are interested in our webinar series. What folks can do actually is quickly, if they have a topic idea off the top of their head, they can use our hashtag on Twitter, which is RegenMedEd. It's pretty simple, REGENMEDED. Or, folks can feel free to reach out to me. We're looking really to make sure that we are supplying information that's needed, and we really want to hear from folks about what they want to learn about from OTAT. So I really appreciate the question. Thanks again.
Lovely, lovely, amazing. The next question would be, where can I find information on past listening sessions and patient-focused drug development meetings?
Oh, sure. So what I would recommend folks do is look on FDA.gov, the Office of Patient Affairs in the Commissioner's Office at FDA. They host our patient listening sessions, and they have an amazing website devoted to the patient listening sessions. And that can show you how to request a listening session. It provides summaries of past listening sessions. And I didn't really go into this detail, but there's a difference between an FDA requested and a patient requested listening session. So the FDA-led listening sessions, those are when our reviewers have specific questions about a disease or a patient experience with perhaps treatments that are available or potential treatments, and they want to kind of deep dive into a particular aspect of their lives. And so those summaries are online from FDA's Office of Patient Affairs. And then there are also brief high-level summaries from patient-requested listening sessions. So you can find them all on the website. I would also encourage you, if you find one a listening session that's a patient-requested session that you find particularly interesting, go to the site for the requesting organization. Go to the patient advocacy organization. They typically will have more detailed information on their requested listening sessions. Yep. So I'm not sure. Did I get to everything you asked, Jen? Just want to be sure.
I think you did. Thank you so much. These are great resources. Again, love the Twitter handle in this age of social media. So thank you, Anne. Up next is Dr. Manuela Sorillo, which would you be able to take the following questions we received, given that the first one addressed to you is, can you provide more details on ways FDA is funding advanced research and development projects to support regulatory science and innovation? And there's a second part to this question. Are there funding opportunities for scientific nonprofits working in the CGT space? Thank you.
Yes, thank you, Jen, for that question. I think that's very important. So we have a website that has a lot of information on the funding opportunities that are available, but also has a list of all the projects that we have supported in the last 3 or 4 years. And you can go to that website. At the end of my presentation, I think it's the last slide has a link to the CBER Advanced Technologies Program. And on that website, you have links to the extramural funding, and also there will be a link to the CAT portal for submitting the request. So that's where you'll find additional information. Now, the second part of the question, whether or not we do fund advanced or R&D research related to CGT, yes, we do. In fact, most of the projects that we have funded are in that space. So I hope that answers the question. Thank you.
No, thank you. Absolutely. We've got quite a few more. So the next one is, can you request a CAT meeting if you have already had an Interact meeting, but not yet had a pre-IND meeting?
So thank you again. If you already had an Interact meeting, that means that you already have a product identified, because as I mentioned in my presentation, Interact meetings are product-specific discussions on CMC, farm talks, and clinical study design. So that would be outside of the scope of CAT meetings, as CAT meetings are really not product-specific discussions. However, if you have a technology that is new and you're applying it more broadly to a product class, that would fit under CAT, but not, product-specific discussions, as I mentioned. Thank you.
No, thank you. Now, the next one is, if a CAT meeting is granted, is there an opportunity to provide additional materials beyond just a slide deck 3 days in advance? With the 2-page limit for meeting requests, it seems difficult to convey enough information for CATs to provide guidance. Would a streamlined meeting package, perhaps maybe less than 20 pages, be welcomed?
Thank you for the question. So the 2-page limit when you submit the request, that was done on purpose to keep the information and as well as the subsequent discussions through the CAT interactions at a high level. So we really are trying to address regulatory implications, regulatory issues that are associated with the implementation of these technologies for manufacturing complex biologics. And so we want to provide just high-level feedback, because if the discussion is too technical or it gets too much into the weeds, we think that other meetings are probably more effective ways of obtaining feedback from the center. These meetings are the Interact meetings or the pre-IND meetings. So, but once a CAT meeting has been granted, as I mentioned during my presentation, you have the opportunity to do a presentation where you can include additional details on that specific technology, to drive the discussion and for information as well. Thank you.
Well, thank you, and that gives me a great segue to the next question, which is when a sponsor is granted a CAT meeting, are these typically held for 60 minutes or 90 minutes, given that they'll be given a presentation?
Yeah, so these meetings are scheduled for 1 hour, and we believe that that's enough to discuss high-level topics that are associated with a specific technology. Now, we do see that initial CAT meeting as a beginning or as the beginning of a dialogue between CBER and the technology developers. So there will be opportunities for subsequent meetings that we can engage in to discuss additional details of the technology. And if not through meetings, we can also continue the dialogue through written feedback via email as well. So it is just the initial interaction is just the beginning of a dialogue, but the CAT meetings are scheduled for 60 minutes. Thank you.
Right. Yes. 60 minutes is short but sweet time amount for it. All right. So then the next question is you kind of touched on this already. Is there an option for follow-up discussion after a meeting with the CATS team, or feedback from the CATS team after a meeting?
Right. So thank you. My answer would be the same. Like I said, the initial CAT interaction is the beginning of a dialogue. So we will follow up through additional meetings or written feedback through email.
Wonderful. Thank you so much. As far as the next question, from the FDA's perspective, what would be the optional time point to discuss a new manufacturing platform with the CAT team? Second part of that question will be early during the concept phase, or rather later once initial proof of concept data is available?
Thank you. That's actually a very good question, and not an easy one to answer, because we do encourage the technology developers to reach out to the center at a very early stage of development, and that's to de-risk any major investments in implementation of that technology for manufacturing complex biologics. But also for us, as I mentioned in the presentation, it gives us an opportunity to become aware and learn about these new approaches, these new technologies. However, if it's too early in development at the concept stage, you may not have a good appreciation of the potential regulatory issues that may be associated with implementation of that technology. So that may be a drawback. Now, if you already done a proof of concept, if it's for a product that's regulated by CBER, then we are already familiar with that technology. So that would be outside of the scope of CAT. Right, but perhaps you have a technology that was applied for the manufacturing of other entities, such as monoclonal antibodies or therapeutic proteins that are regulated by CDER, and you want to use that technology to manufacture more complex biologics that are regulated by CBER, then in that case, a CAT meeting would be suitable. Thank you.
Great answer. Thank you so much. And I have two more questions that could be kind of yes or no. The first one is, are CAT meetings also available for CDER products?
No, CDER has the emerging technologies team, which can address questions related to those technologies.
Right. Thank you so much. And then the last one is it best to request and have a CATS meeting before an Interact?
No. Well, it depends on where you are in what stage of development you are, right? If you have the technology and you don't have a product identified, then CAT would be ideal. But if you already have a product, then you will get perhaps more feedback through an Interact meeting where you can discuss product-specific regulatory issues. Thank you.
Thank you so much, Manuel, for your time on those questions. Next up is Crystal. Please, Crystal, would you address the following question to you? Can you please advise regarding the timelines for the PSA meeting request and meeting package submission? Should the PSA meeting request and meeting package be submitted at the same time, or is there some time after the meeting has been granted to submit the meeting package?
Thank you for that question. The timelines for the PSA meeting request could be at any time, and the meeting package does not have to be submitted at the same time as the request. So the request should come in separately. Then after the request is either granted or denied, if it's granted, then there will be a discussion about when is a good time for the sponsor to submit, but there is no exact time frame between the meeting request and the meeting package to be submitted, and not submitted at the same time.
Thank you so much. The next question to you would be, does the sponsor participate in the debriefing discussion following the trilateral meeting?
No, thank you for that question. The debriefing discussion following the trilateral meeting includes EMA and FDA and 2 patient representatives if agreed upon by the agencies and the sponsor. The debriefing discussion gives FDA and EMA an opportunity to discuss the responses to the agenda items, follow-up questions or concerns, and any discussion on future plans or submissions of the product. Thank you.
Wonderful, thank you. Next question is, will there be one single point of contact for sponsors during the PSA meeting process?
That's a good question. The sponsor will have 2 point of contacts, the project manager from FDA and another point of contact from EMA.
Awesome, thank you so much. The next question is, what is the best way to prepare for the trilateral meeting with respect to receiving the list of issues from EMA and FDA preliminary comments separately?
That's a great question to prepare from the sponsor's point of view. EMA will send the list of issues to the sponsor 7 days prior to the trilateral meeting, and FDA will send the preliminary responses 2 days prior to the trilateral meeting. FDA requests that the agenda and the order of questions 1 day prior to the meeting. So the sponsors should prepare their questions for the trilateral meeting in order of importance, listing which questions are for FDA, EMA, or both. Taking the timeline into consideration, the sponsors should plan accordingly for that timeline because they will be coming in separately. Thank you.
No, thank you. A couple more. During the trilateral meeting, who will be the moderator?
Thank you for that question. During the trilateral meeting, FDA and EMA will start with a brief introduction of the submission, as well as some background information about PSA meetings. Both agencies will give introductions of their core review teams, and the sponsor participant introductions will follow. The meeting will then be turned over to the sponsor to moderate for discussion of the prioritized questions that they are seeking further clarifications for.
Awesome, thank you, Crystal. I think those are all your questions. I would like to circle back to Anne, if I may, for one last question as we have a few minutes left. The question to you, Anne, is how does FDA recruit patient participants for the listening sessions?
That's a really good question, Jen. Thanks to the audience for picking up on that. Something I may or may not have mentioned in my earlier answer is that FDA has partnered with the National Organization for Rare Disorders in its patient listening session program. So for an FDA requested listening session, and you'll remember from my earlier discussion that these are questions where listening sessions where FDA has some specific questions for patients living with a particular disease or condition. And once those questions have been identified, the review division reaches out to the Office of Patient Affairs, and they reach out to our contacts at NORD, who are partnering with us for these listening sessions. And the folks at NORD will then reach out through their contacts and their member organizations and help to identify patients or caregivers, depending on the type of questions and the disease the disease in question, and they will help recruit patients that can participate in these listening sessions. So for the FDA requested sessions, they're usually about an hour and a half. They're about 7 or 8 questions that our reviewers may have, and it's really a discussion with each of the patients or caregivers who've been identified to hear their perspectives on these different questions. Then again, when it comes to a patient-requested listening session, those are coming into us directly from the patient organization. So hopefully that helps.
Well, that's very, very good and very interesting to hear about. And with that, I'll have to end our Q&A session on that note, as these are all the questions that came in. But again, a big thank you to Dr. Anne Rousey, Dr. Manuel Osorio, and Crystal Melendez for your time and expertise. So this concludes this session and leads us into our afternoon break. And we will return at 3 p.m. Eastern Time with our next triple header presentations of medical devices in CBER, safety-related impurities in plasma-derived products, and common mistakes in demonstrating analytical method suitability, all moderated by our Dr. Graham Price.
Good afternoon and welcome. I'm Dr. Graham Price and I'm a gene therapy reviewer in CBER's Division of Cellular and Gene Therapies. But this afternoon I'll be serving as the moderator for this final session of the conference. We will have presentation on quite diverse topics from 3 CBER speakers, followed by a joint question and answer session. It's my pleasure and privilege to introduce our speakers. Dr. Alyssa Kitchell will be speaking on medical devices in CBER. Dr. Kitchell received her BS in Chemical Engineering from Tufts University and a PhD in Biomedical Engineering from Georgia Tech and Emory University. She currently serves as Device Team Lead in the Tissue Engineering branch in the Division of Cellular and Gene Therapies. Dr. Mikhail Avanasov is a principal investigator in the Division of Plasma Protein Therapeutics and will be speaking about safety-related impurities in plasma-derived products. His laboratory studies the safety and efficacy of hemostasis products, and he reviews regulatory submissions for a variety of plasma-derived and recombinant plasma protein therapeutics. Dr. Emnat Barak will be presenting on common mistakes in demonstrating analytical method suitability. He received his PhD in chemistry from North Carolina State University and worked in industry prior to joining CBER's Division of Biological Standards and Quality Control, where he evaluates the suitability of analytical test methods in biologics license applications and is involved in the lot release program for CBER-regulated products. Please remember to type your questions into the Q&A pod and address them to the particular speaker. Without any further ado, I'll turn the floor over to our first presentation.
Thank you for that introduction. I'm very pleased to be able to talk to you today about medical devices reviewed in CBER, the Center for Biologics Evaluation and Research, as well as performance considerations for delivery devices of biological products. My goal is that by the end of this talk, you'll be able to identify the product offices in CBER that review medical devices, and identify the types of medical devices CBER reviews, and describe delivery device considerations for biological products. In the first part of my talk, I'll go over the types of medical devices reviewed in CBER. So when we think about medical devices at FDA, it makes sense that the Center for Devices and Radiological Health come to mind first. But there are certain types of medical devices that are actually under CBER's jurisdiction, which we'll talk about more in a few slides. First, I want to note that even though there are differences between the types of devices that CDRH and CBER regulate, there's actually a lot of overlap in the types of device submissions that the two centers receive. Third-party pre-market notifications and CLIA categorizations are specific to CDRH, but other than that, CBER reviews all the same types of device submissions as CDRH, including Q submissions, IDEs, 510Ks, de novos, PMAs, HDEs, and 513Gs. As you might be able to tell by that list, that means that both CDRH and CBER regulate Class 1, Class 2, and Class 3 devices. Other types of submissions that CBER receives are pre-submissions, such as Interact and pre-IND meeting requests, INDs, BLAs, NDAs, and ANDAs. Some of these submissions may also include delivery devices, which I'll talk about in the next part of my talk. CBER also reviews a hybrid type of submission called device BLAs, which are for in vitro diagnostics, such as donor screening assays that are both devices and biological products, as explained in 21 CFR 809.3. and Section 351 of the PHS Act. Even though the submission is a BLA, the application is subject to MDUFA fees and timelines. We'll see a few examples of device BLAs in the coming slides. Now that we've covered the topics of device submissions that CBER reviews, we'll identify the product offices within CBER that review medical devices. Of the product offices in CBER, devices are primarily reviewed by the Office of Tissues and Advanced Therapies, OTAT, and the Office of Blood Research and Review, OBRR. We'll start our overview of the types of devices reviewed by CBER with the devices in OTAT, which is my home office. From a general point of view, CBER-OTAT reviews devices that contact human cells, tissues, and cellular and tissue-based products, HCTPs, and some blood products in some way. That includes devices that collect or process or store HCTPs for a therapeutic purpose, such as cord blood processing devices and hematopoietic. Within the category of processing are also devices that process HCTPs ex vivo at the point of care, where the therapeutic device is mediated by the device output. Devices in this category are typically Class 3 devices that require an HDE or PMA. For example, many of you might be familiar with the Clinimax CD34 reagent system, approved under an HDE, which uses antibody-conjugated magnetic beads, a buffer, a tubing set, and an instrument to enrich CD34-positive hematopoietic progenitor cells from a donated apheresis product for use in patients with acute myeloid leukemia undergoing allogeneic stem cell transplantation. Another example is the RECELL autologous cell harvesting system, approved under a PMA, which creates an autologous cell suspension from harvested skin at the point of care to spray onto a larger area of burn wounds. CBER-OTAT also reviews devices that process peripheral blood to create platelet-rich plasma, PRP, or platelet-rich fibrin intended for a therapeutic purpose. For example, we have devices that generate PRP at the point of care from mixing with allograft or autograft bone before application to a bony defect to improve bone graft handling properties, and some PRP devices where a PRP gel is generated and topically applied for management of exuding cutaneous wounds. These examples are Class 2 devices that require a 510K pre-market notification, but other devices in this category could be Class 3 requiring a PMA depending on the indication. They might also be under CDRH jurisdiction if they have an in vitro diagnostic intended use. So speaking of in vitro diagnostics, CBER-OTAT also reviews certain in vitro diagnostics that are intended to count cell populations in HCTPs, such as these image-based and flow-based assays for CD34 positive cell enumeration. Some of the most interesting devices that CBER-OTAT reviews, in my opinion, are device constituents of certain biologic-led combination products. These include things like biomaterial or synthetic scaffolds that are combined with cells to make tissue-engineered constructs for regenerative medicine. An example of this is the porcine collagen membrane that's combined with autologous cultured chondrocytes to make a graft for repairing symptomatic cartilage defects in the knee. We also review certain devices used to administer biologics, such as pre-filled syringes, which are single-entity combination products, and devices that are co-packaged or cross-labeled to be used with a specific biologic. So at this point, we've covered the types of devices reviewed by CBER-OTAT. Now we'll take a look at the types of devices reviewed in OBRR. As you might guess, the Office of Blood reviews devices that are used in establishments that manufacture blood or blood products. Within OBRR, there's two different divisions that review devices in this category. The Division of Blood Components and Devices, DBCD, and the Division of Emerging and Transfusion Transmitted Diseases, DETTD. First, we'll take a look at the devices reviewed in DBCD, which are very diverse. If you've ever donated blood or plasma, you might be familiar with the blood or blood component collection devices or other devices used to test, process, collect, and store donated blood and blood components, such as apheresis machines like the one pictured here. It's also worth noting that some of other uses of apheresis machines are reviewed in CDRH. Some immunohematology in vitro diagnostics are also reviewed by this group, including HLA kits, blood grouping reagents, reagent red blood cells, and anti-human globulins. An example is the reagent red blood cells approved under this BLA, device BLA, that are used with a 510K cleared automated blood grouping and antibody test system. Outside of reagents, DBCD also reviews devices intended for use in preparation of, in conjunction with, or for quality assurance of a blood bank-related licensed product or practice. This includes devices like blood establishment computer software, devices with blood irradiators, and blood product thawing devices such as this plasma warming device pictured here. Finally, to wrap up the devices in OBRR division, we have devices and combination products that are used to prepare, preserve, and store blood products, including bacterial detection devices and pathogen reduction systems, such as this PMA-approved plasma irradiation system. As you might be able to gather from the name, the types of devices reviewed by OBRR DETTD focuses on in vitro diagnostics. These in vitro diagnostics include donor screening assays and multiplexes for infectious agents which are reviewed under device BLAs. For example, Alinity S HTLV antigen with Alinity S system, and retroviral diagnostic tests such as the OraSure in-home HIV test, which are approved as PMAs. For the next part, I'll discuss delivery devices for biological products. If we want to get a biological product into a patient, what do we need? A delivery device. Selecting what type of device depends on where and what the therapeutic target area is. Some examples of delivery devices are a syringe and needle, jet injector, microneedle patch, and an intrabronchial spray device. Please note that FDA considers scaffolds, wound coverings, or encapsulation materials to impart more than a delivery function. Along with thinking about how we want to get the therapeutic agent into the patient, there are several other points to consider as we choose a delivery device for a biological product. The safety and effectiveness of the delivery device is critical for the safety and effectiveness of the therapeutic. The sponsor of the therapeutic determines the performance requirements of the delivery device, and most biological product submissions should identify delivery devices for their therapeutic. Typically, a biological product that requires a delivery device is considered a combination product as defined in the regulations. If it is provided as a single entity, or if it is co-packaged or packaged separately but cross-labeled for use with an approved product or an investigational product. When we receive a biological product submission that identifies the use of a delivery device, we review a variety of information to support the safety and effectiveness of the delivery device. As you can see, there's numerous types of information needed to determine the safety and effectiveness of a delivery device. For today's talk, I will only be focusing on specific considerations related to device performance. Performance testing for the delivery device should assess specific functional requirements, also termed design verification, and also ensure that the device achieves its user needs and intended use, termed design validation. Such verification and validation could be bench tests, could be in vivo preclinical animal testing, and/or clinical studies. The performance metrics assessed during verification and validation should also be included in the overall final product stability program. It is important for the therapeutic manufacturer to identify and control the critical design outputs needed to deliver the intended product dose to the intended delivery site, starting from product preparation and delivery dose initiation until completion. Additional performance testing should include examination of the device with the biological product to ensure product quality and delivery device performance are maintained. Such assessments should use the worst-case clinical parameters. So let's quickly summarize what we've covered. Overall, for this presentation, we covered the medical devices reviewed in CBER, ones used in point-of-care manufacturing of HCTPs, devices used in blood establishments, and combination products where there are device constituents in biological lead products. As for delivery devices, those should undergo design verification and validation as part of their performance testing. And additional compatibility testing with the final biological product is key. Now let's test your knowledge with some challenge questions. For the first question, let's consider a manufacturer that intends to market a device in the United States that collects and stores cord blood. What group in FDA will review their submission? A, CDRH, B, CBER, Office of Blood Research and Review, C, CBER, Office of Tissues and Advanced Therapies, or D, CBER, Office of Vaccine Research and Review. If you answered C, CBER OTAT, you are correct, since this device is intended to collect and store cord blood, which is an HCTP. For the second question, identify which of the following statements are not true. A, some devices that deliver or administer biologics are reviewed by CBER. B, in vitro diagnostics intended to diagnose HIV are reviewed by CBER in the Office of Blood Research and Review. C, blood establishment computer software is considered a medical device and reviewed by CBER in the Office of Tissues and Advanced Therapies. Or D, an IDE to study the use of bone marrow concentrate generated by a device at the point of care to treat osteoarthritis would be reviewed by CBER in the Office of Tissues and Advanced Therapies. The statement that is not true is C. Blood establishment computer software is indeed considered a device, but it is reviewed in CBER OBRR since it would be used in an establishment that manufactures blood and blood products. And for our last question, let's fill in the blank. What is testing to ensure the delivery device meets specified performance or functional requirements? A, design verification. B, combination product. C, design validation. D, shelf life. If you chose A, design verification, great job. And with that, I'd like to direct you to these additional resources and ways to contact us. Thank you for your attention.
Hello, my name is Mikhail Ovanetsov. I'm a research biologist and principal investigator on a research program in the Division of Plasma Protein Therapeutics of the Office of Tissues and Advanced Therapies, CBER. The topic of my presentation is safety-related impurities in plasma-derived products. Our learning objectives for today are to describe examples of impactful investigations into unexpected serious adverse events caused by impurities and contaminants in plasma-derived products, and to discuss manufacturing root causes and mitigation strategies developed in response to these adverse events and investigations into these adverse events. I need to start with a disclaimer. My comments are an informal communication and represent my own best judgment. These comments do not bind or obligate the Food and Drug Administration. So what are plasma-derived products? These are the products derived from blood plasma. Human blood plasma is a complex biological fluid rich in proteins, carbohydrates, and lipids. But the majority of plasma-derived products are concentrates of blood plasma proteins. For example, immune globulins, normal immune globulins purified from large pools of human plasma derived from normal donors, or hyperimmune immune globulins purified from plasma of patients or donors exposed to certain pathogens, for example, hyperimmune, anti-SNAC venom immunoglobulins. Also have albumin, coagulation or blood clotting factors, and topical hemostasis agents, for example, fibrin glue, thrombin spray. Now, plasma protein therapeutics are biological products, and all biological products contain impurities because complete removal of process and product-related impurities is impossible. Impurities in general are potential critical quality attributes, or CQAs, because of their potential impact on product safety. This is according to ICH Q11. And control strategy should consider both manufacturing feasibility and safety risks. Obviously, we need to apply greater concern to those impurities that have confirmed impact on product safety. Fortunately, cases of product safety impact are quite rare. This is true to impurities in plasma-derived products as well. This is because plasma components and plasma proteins can co-purify with target proteins. When we purify, for example, albumin, we would purify many other proteins along with the albumin, but not all of them would be present in high enough concentrations to have a biological activity in the patient population. In fact, some of the impurities in plasma protein therapeutics can be viewed as harmless. They would provide protection for the target protein from the harmful effects of purification or in the final formulation of the product. However, select few impurities and contaminants were associated with clusters of serious adverse events in the past, such as hypertension, viral transmission, bleeding, and thrombosis, and this is what we're going to talk about today. Real confirmed cases of safety impact caused by real impurities. We will start with the hypertension, a case of multiple adverse events recorded in the beginning of 1977. All cases that I'm going to discuss today are presented in the chronological order, and let's start with hypertension. These cases were recorded for the product called plasma protein fraction, or PPF, which was available since 1950s. It is still available today, part of CFR Part 640. This is a protein fraction comprised of 85% pure albumin, and it is heat-treated to inactivate hepatitis B virus. The reason why heat treatment is important, because just several years prior to 1977, there was a well-publicized cluster of hepatitis B transmission in patients who received plasma protein fraction. Since then, or even earlier than that, all PPF is inactivated with heat treatment to remove hepatitis B virus. This product was widely used between 1950s and 1980s as a colloid volume expander. It is still given today in critical blood loss situations to avert hypertension. Although, and this is another disclaimer, no solid evidence exists on the benefit of this product versus crystalloids. Now, the reason why this product is used is because it can reduce hypertension. A patient has reduced blood pressure, receives a dose of PPF, and blood pressure goes up. Blood pressure is normalized. It's important that physicians can monitor blood pressure in the patient very accurately in real time, so they see that PPF is actually working. In 1977, they've seen the opposite. Patients had reduced blood pressure, they received certain lots of PPF, and blood pressure dropped even further. Patients were at risk of severe harm, and clinicians were very concerned. FDA received these lots, received reports of these events associated with the product. They received the lots, and this paper here describes the results of the scientific investigation into the root causes for these hypertension events. It was published in 1978, full year after the cases were fully investigated and actions were taken to improve the safety of the product. But it all started with just 13 lots of PPF product made by just one manufacturer, which were implicated in 23 reports of hypertension in surgical patients. What they found at the FDA, in the FDA lab, was the presence of high levels of impurity called prekallikrein activator. This is actually coagulation factor 12A beta, which is a small protein that activates prekallikrein. It can be present in plasma in relatively low concentrations, but its levels were very high in these product lots that caused adverse events. FDA lab developed a method and a reference standard to measure these impurities, and they were able to confirm, together with additional evidence of additional lots and additional adverse events, they were able to connect the levels of impurity with actual adverse events. 9 nanogram per ml in the product can cause a hypertensive episode, and 25 nanograms per ml can cause total circulatory collapse. The story presented here is described in a document that you can find on the website of the Food and Drug Administration, and that investigation resulted in a huge recall. In late May or early June of 1977, just several months after the events were received by the Food and Drug Administration, 1.2 million grams of protein or 25 kilotons of product, and this is usually a 5% solution, was recalled. Huge recall. The reason why this recall was fully justified at that time was not only that there was a confirmation that pKa was present in these lots of the product, but that the elevation in pKa was caused by the manufacturing change that was not fully described by the manufacturer, which has caused the increase in pKa in the product. So there was a change that caused an increase in pKa, and the increase in pKa caused multiple adverse events. But it went even further. Since that time, since we have now solid evidence of the association of high pKa levels in the product with adverse events, all lots of albumin, PPF, and immune globulin products are now tested for the presence of pKa impurity. We have different levels for albumin, PPF, and immune globulins, depending on how these products are used and how these patients are affected by the pKa. But since 1970s, all lots of these products are tested for pKa. The companies have to develop pKa methods for release. There are international reference materials produced to standardize these methods, and the requirements are harmonized between the European Union, for example, and the Food and Drug Administration. It was a dramatic shift for the whole plasma purification industry. Now, another case, you've probably heard about it. It all started in 1982. Case of viral transmission in the hemophilia population. Hemophilia is a bleeding disorder caused by the deficiency of coagulation factor 8, hemophilia A, or coagulation factor 9, hemophilia B. Prior to the introduction of plasma concentrates or stored plasma, the life expectancy of hemophilia patients was very low. 10 years, average life expectancy was about 10 years at the beginning of the century, and it was increased to 70 years in the decade prior to these adverse events. In 1982, factor concentrates, which are prepared from large pools of plasma, multiple donations of healthy donors, would be pulled together, treated through a purification fractionation process to prepare concentrates of coagulation factors of interest. They've been found to be the source of AIDS infections in hemophilia patients. These pools were contaminated with the HIV. In just 2 years, 60% of US hemophilia patients were infected with the HIV via the concentrates. And as a result, the life expectancy in this community has dropped to just 40 years in the decade after the introduction of viruses into the population. Today, we still use plasma-derived products and plasma-derived concentrates. They are safe and effective. The risk of viral transmission has been effectively, practically fully, in this community, mitigated via the qualification of plasma donors. Now all donors are tested for several known viruses. The donations are kept by the blood collection centers and tested when the donor comes over for the second time. We use reliable viral tests that were developed specifically for this purpose, and we use them both on the individual donations of plasma and on the manufacturing pools. We also use orthogonal viral inactivation and reduction process steps to inactivate known and unknown viruses that may be present in the pools of plasma. But it is important to note that these impurities, these viruses, are actually contaminants in a product because they were adventitiously introduced through materials, through plasma, and they were not intended to be part of the manufacturing process. Pools of normal plasma are collected from healthy donors. They are not collected from known infected individuals. Another important consideration here is that there are multiple products on the market now, and there were multiple plasma protein concentrates on the market back then. And it was the hemophilia community that was affected by this particular virus because this virus happened to co-purify with the blood coagulation factors. In the end, it was the manufacturing root cause that contributed to the increased risk of viral transmission in these patients. And that was accounted for in the development of improved manufacturing processes and improved control strategies for the factor concentrates that have been introduced in response to these adverse events. Now, another case has been first reported in 1989 And this is a case or a number of adverse events that were caused by acquired bleeding disorder. They've been reported to a product called topical bovine thrombin. Thrombin is a blood coagulation enzyme that converts soluble fibrinogen, a protein that circulates in plasma, into fibrin clot, into a fibrin that spontaneously forms fibrin clot, which is the principal component of the thrombus or hemostatic plaque. This particular thrombin product was derived from bovine blood, bovine plasma, and used topically as an adjunct to surgical hemostasis since as early as 1940s. It was FDA approved since 1970s, and in late 1980s, we've received reports of acquired bleeding conditions that were linked to product exposure during surgery. Initially, the root causes were not fully understood. We knew that adverse events included a number of problems: impaired food repair, other situations, other events, thrombosis, uncontrolled bleeding, shock, or even death. And eventually the root cause was identified as patients' own antibodies raised against bovine factor V that were cross-reacting with human factor V. It was a case of acquired factor V deficiency. And the root cause for that was the impurity factor V fragments in thrombin concentrates. Thrombin does not circulate in blood plasma. All enzymes, all coagulation enzymes, are quickly inhibited by plasma inhibitors because we don't want blood clotting to happen anywhere where it doesn't need to happen. Thrombin is produced naturally at the site of vascular wall injury, or it can be produced in the manufacturing process. When it is produced in the manufacturing process, different impurities can be produced as well, and that's what happened at that time in the early manufacturing processes for the bovine thrombin, and the manufacturing processes were modified to reduce the number of these impurities, and the risks are effectively mitigated right now. The amount of these impurities is controlled, the manufacturing processes were modified. Now, the last case of adverse events is related to thrombosis with another product, immune globulin intravenous. Immune globulin intravenous, or IGIV, is a concentrate of mostly IgGs. They've been available since 1940s. The first IV formulation product was introduced in 1981, and they are manufactured from large pools of plasma, from 7,000 donors usually to 60,000 donors would contribute plasma to these pools. There are multiple indications: immune deficiencies, Kawasaki syndrome, various neurological, blood, skin, many other diseases. This is actually a very important product. It is estimated that the majority of blood plasma collected for further manufacturing use would end up being fractionated into immune globulin products. Several years after the introduction of the intravenous formulation of this product, first literature reports of thrombotic events were published. In early 2000s, FDA performed an investigation into the rates of spontaneously reported thrombotic adverse events for these products, and we've been receiving about 30 events per year. These cases are very severe: myocardial infarction, stroke, deep venous thrombosis, and pulmonary embolism, to name a few. and therefore precautionary labeling has been recommended by the Food and Drug Administration for IGIV since 2003. But back then, cases remained uncertain. This is because there are theoretically different explanations to thrombotic adverse events. For example, this is a highly concentrated protein solution that is infused in large volumes. That alone, the viscosity alone, can theoretically cause disturbances that may translate into rare thrombotic adverse events. Other explanations were that some of the patients had underlying conditions that were actually responsible for these thrombotic adverse events, not the product. They would have developed the thrombotic events even if they were not to receive IGIV therapy. That was the explanation. All has changed in 2010 when a manufacturer reported a cluster of thrombotic events associated with 2 product lots. Our lab, my lab at the Food and Drug Administration, received these lots along with non-implicated product lots. And we have found coagulation factor 11A impurity, coagulation enzyme, in the affected product lots. And we have proposed a detection method, which we have shared with the company that manufactured this product. They've confirmed our finding. They found many lots that had high levels of factor 11 impurity. They also found that more lots were associated with thrombotic events. And as a result of these investigations, many lots of the product were withdrawn from the market, and later on, when we received even more adverse event reports, all product lots were removed from the market. In 2010-2012, Food and Drug Administration and European agencies engaged in an industry-wide evaluation of impurities and adverse events. We've conducted pharmacovigilance studies, and we have found more products potentially involved in thrombotic adverse events, more products with impurities, and a number of product lots were removed from the market. As a result of these investigations, manufacturers of all investigational and most licensed products agreed to introduce manufacturing changes to reduce factor 11A impurities and lot release testing to control factor 11 impurity levels in the product. Some products were removed from the market entirely, others have changed indications to reduce the risk of thrombosis in these patients. We've also started working on the harmonization of release testing methods and limits. This work is still ongoing. It started with 2011, a public workshop on risk mitigation organized by the Food and Drug Administration. International standards for coagulation factor 11 impurity were developed by the World Health Organization. European organizations started working on the common test. Industry contributed to that discussion, and currently Food and Drug Administration regulatory agencies in Europe are working to develop harmonized methods. The task is significantly more complicated than we thought, and the harmonization is still ongoing. Now, that was the last slide of my presentation, and we are going to challenge questions. Challenge question number 1. Which of the following is a contaminant? PKA enzyme, HIV, factor 5 fragments, factor 11A enzyme. And the answer is HIV. HIV is a contaminant. It wasn't expected to be in the final product at all. Challenge question number 2. Which of the impurities or contaminants were not expected to come from plasma? Coagulation enzymes, PKA and factor 11A, bovine factor 5 fragments, viruses, or none, i.e. all were theoretically possible. The answer is all were theoretically possible, human plasma in general can be a source of all of the above. So in summary, plasma-derived impurities and contaminants have caused severe adverse events. We know it from our history. Plasma-derived products can be dangerous if manufactured improperly. Post-event mitigation strategies have been introduced and they have been successful, but they can be very burdensome to the industry. Product recalls, new process steps developed, validated, and introduced, new in-process and release testing is introduced. Overall, severe adverse events that I have described were caused by theoretically known, although not yet controlled impurities. We didn't discover anything absolutely new. We knew about the viruses, we knew about coagulation enzymes, we know how to detect them. Maybe our methods were not good enough for the quality control environment, but scientifically they were known impurities and contaminants, but they have caused adverse events. And my closing thought, sponsors should consider relevant product-specific impurities and contaminants and possible safety risks. While the examples that I have described today are relevant to plasma derivatives industry, there are multiple other examples for other products, or even similar examples for other products that can be cited when we're talking about safety impact. And there are theoretical considerations. For example, coagulation factors are present in serum, which is used to culture and produce cell-based therapies. Coagulation factors are expressed by some of the commonly used cell substrates in the recombinant protein manufacture. Whether your process will enrich these impurities and whether you can control these impurities would define the safety of your product. Thank you very much, and I will be happy to answer your question.
Hello everyone, and thanks for staying for this presentation. My name is Imnatit Barak, and today I will briefly talk on method suitability and validation. I will also provide some real-world examples of common validation mistakes that we see during regulatory reviews. So the learning objectives for today are to describe the FDA regulations related to product testing, plus why and how test method suitability is evaluated. Identify the current validation requirements from FDA, ICH, and USB guidances. Identify different method types and how method performance is evaluated during validation. Provide examples of some common issues or mistakes in method validation that we observe in regulatory submissions during our review. So what are the federal rules coded in 21 CFR regarding the need for validation or evaluation of suitability before test methods are used for product testing? So here are some direct quotes from 21 CFR. First one is from section 211-165E. The accuracy, sensitivity, specificity, and reproducibility of test methods employed by the firm shall be established and documented. Section 211-194A2, the suitability of all test methods used shall be verified under actual conditions of use. So what are the validation expectations according to the guidance documents for the different types of analytical methods to demonstrate their suitability. You can see there are 4 different departments of different analytical procedures or methods, and the parameters are along the first column you see here. Evaluation of all parameters may not be expected for all method types, except specificity is required by all method types, and also robustness is required by all methods. However, robustness is usually performed early in the method development and optimization stage, usually not during validation, but the robustness data should be submitted together with the validation package. The level of validation required also depends whether a method is compendial or not. Compendial methods are standard methods described in the chapters of the United States Pharmacopoeia, USP, or other regional compendia like European and Japanese Pharmacopoeia. Only verification of the method is required for compendial methods, and for verification follow USP General Chapter 1226. Verification suitability should be under the actual conditions of use. That means using the actual drug substance or drug product, not just using a standard. Non-compendial are methods that are not in the pharmacopoeia. Usually this includes methods that are developed in-house. For validation of non-compendia, follow ICH Q2R1 for validation. Identify and assess the appropriate validation characteristics that are appropriate for the method types. If the method is stability indicating method, there are some extra steps required for specificity. Analyze specificity in the presence of all actual and potential degradants. Potential degradants are degradants that probably arise during stability or during shelf life. If for potential degradants, you may use physical and chemical stress conditions like agitation, heat, light, humidity, etc. to create potential degradants to assess specificity and also the other relevant validation parameters. Here is one common mistake for compendial method validation that we see sometimes. So people claim there is no need to generate verification data before using a compendial method for release testing. For compendial methods that are read alone, like pH and osmolarity and the like, you must verify or show repeatability prior to using the method for testing. You must also prepare the SOP, which should be customized to the product and the instrumentation used. Compendial methods that require culture or further analysis, like sterility and endotoxin, the USP method should be followed. If not followed, the method is considered an alternative method and should be fully validated with data provided to demonstrate sensitivity equivalent or greater than the compendial method. Data should also be provided to demonstrate the matrix, which is a non-active ingredient of the drug, does not interfere with the measurement. Absence of signal enhancement or inhibition, which is interference, is demonstrated by an acceptable recovery of the positive control. If there is a signal enhancement or inhibition, meaning interference, sample may need additional dilution or further washing steps. So here are some general considerations and common mistakes observed during validation. To execute a method validation, an approved written protocol should be followed. Some people probably just go ahead with validation without following a protocol. Another common mistake, a written protocol was not used, as I mentioned earlier, or was not followed if there was a validation protocol, or validation acceptance criteria were not provided, or they were poorly defined in the protocol. The procedure or the SOP describing the method to be validated must be followed. So everything, including the number of replicates, calculations, strictly should be followed according to the SOP during the validation, unless otherwise is justified. This will be justified when you perform accuracy study using spiking, or when you are using an orthogonal method for verification of accuracy. Another common mistake is method is not adequately described in the regulatory filing. So this is something we also see. The procedure is not provided adequately for us to be able to follow the method to execute or duplicate or replicate the method in-house. An actual product was not used in the validation, so sometimes we see standards are used instead of real samples, real drug substance or drug product samples for validation. So validation studies should be conducted with representative sample, active ingredients plus the matrix used in the test of the drug substance or the drug product. So during the test of using the method, if drug substance and drug product are used, then for validation the same materials should be used. Another common mistake is concentration of samples used in the validation did not cover the specification range. So during spiking for accuracy study, spiking of the standard into a drug substance or drug product sample for accuracy study should not alter sample characteristics significantly. So it's advisable to spike small microliter volumes from a high concentration stock standard instead of spiking larger volumes, which may change the behavior of the sample. So our first challenge question, the accuracy, sensitivity, specificity, and reproducibility of test methods employed by the firm shall be established and documented. This statement is directly taken from A, ICHQ2, B, 21 CFR, C, USP 1225, D, Einstein's lab notebook. So if your answer is B, then you are correct. So let me briefly talk about each validation or performance characteristics, starting with specificity. The ICH definition for specificity is the ability to assess unequivocally the analyte in the presence of components which may be expected to be present. Specificity is required for all types of analytical procedures. Specificity should show, as you can see on the figure on the first chromatogram, the absence of matrix or placebo interference, including suppression or enhancement of the response by the matrix. So this is an overlay which shows the matrix does not have any signals that are the same as the drug substance or the drug product. Common mistake that we see, not submitting a chromatogram overlay as shown in figure 1. So you need to provide such an overlay to demonstrate absence of matrix interference on the measurement of the drug. Resolution. Resolution is one quality of specificity. It's the ability to resolve an analyte from other components. Hence, the chromatogram shown in Figure 2 must be provided to demonstrate specificity if there are multiple components. So the start component that you see in the non-specific method, everything is co-eluding, so this method would be non-specific, while the specific method on the right side has resolved the start analyte component from the other two components. In case you have a single peak profile, then you need to perform peak purity analysis to show that that single peak is purely single. There are no extra peaks hidden underneath an apparent single peak. So here is a typical common mistake we see on specificity. This example is from an actual BLA submission. This HPLC method was submitted to determine identity and percent composition of a product and the impurities. As seen on the chromatogram, the product elutes before the green line, and the impurities elute after the green line. And then there is no sufficient resolution between the product and the impurity you can see here. Hence, therefore, this method cannot reliably identify or determine the percent of the product. By definition, as you can see from ICH, specificity is the ability to assess unequivocally the analyte in the presence of components which may be expected to be present. Hence, therefore, this method is not suitable for use, so it's not approvable. So this method may need further development and optimization before an actual use for sample analysis or testing of actual samples. The next parameter is linearity. The ICH definition for linearity is the linearity of an analytical procedure is its ability to obtain test results which are directly proportional to the concentration of analyte in the sample within a given range. Linearity should be evaluated with a plot of signals as a function of analyte concentration or content or percent composition. That means plot the amount the amount reportable on the x-axis. If it's percent composition or concentration, it would be on the x-axis, versus on the y-axis, it will be the instrument response. A minimum of 5 concentration levels are recommended to determine linearity. Common mistake actually that we see is demonstration of linearity using a plot of expected concentration versus measured concentration. This type of plot used for linearity, which is a mistake. We see this commonly in regulatory submissions. However, such correlation is a measure of accuracy, which is a correlation between the measured value and the expected value. Another common mistake that we see for linearity and actually probably applies in accuracy also standards are used to demonstrate linearity. Suitability of the method should be demonstrated with the product itself for linearity. The slope between the sample and the standard curve could be different due to matrix differences. So the standard doesn't have the matrix, but the sample has the matrix. So the analyte in the response of the analyte in the standard could be different than the standard which is diluted in a buffer or water. So the sample concentration determined using a standard curve could be lower than the actual concentration due to matrix. So if the concentration of a sample is determined using a standard curve without the matrix, the concentration determined could be different or lower. Need matrix matching of standards and samples. But this issue may not be significant for separation methods. So for methods like HPLC, this may not be an issue of the matrix effect because the analyte is resolved out by the HPLC before it's detected. But this still needs to be demonstrated that there's no matrix effect. Even in HPLC, you need to demonstrate that. Note this is also a common problem, as I said, when determining method accuracy. Lower or higher spike recovery can happen due to matrix effect. Common mistake for linearity that we see is for incorrect demonstration of linearity for molecular size distribution methods like size exclusion chromatography. So what you see here is a chromatogram, a size distribution by size exclusion chromatography of the different components in a protein IgG sample. The product or the purity is determined from the monomer. So the others, the dimer, polymer, aggregate, and fragment are impurities. This is a critical QC test for protein therapeutics like factor VIII, IgG, antibodies, and oligo-based products like DNA and RNA-based drugs. This is a purity and impurity method to determine the proportion of product, which in this case, as I said, is the monomer. And the product impurities are the aggregates, the dimers, and the fragments. And they are reported using the percent peak area. So the percent peak area of the monomer will be its percent composition. So as I said, the reportable value is the percent peak area or the percent composition. So it's not possible to determine linearity using concentration versus response plot. So linearity should be demonstrated by plotting percent peak area, which is on the y-axis here, which is the reportable value, versus the peak area, which is the response on the y-axis. So such linearity curve should be provided to evaluate linearity for each of the reportable components. If aggregates and polymers and fragments are reportable values, and each should have demonstrated linearity with such a type of curve, including the product. This way of assessing linearity applies to similar methods that report results as a percentage or proportion. Example, as I said, is purity/impurity methods for protein and oligo-based therapeutics. Accuracy. The ICH definition for accuracy of analytical procedure expresses the closeness of agreement between the value which is accepted, either a conventional true value or accepted reference value, and the value found. Accuracy evaluates the closeness, the correctness of the method. Demonstration of accuracy is not required for qualitative or ID methods and limit test methods. Limit test methods are usually semi-quantitative tests used to identify and control trace quantities of impurity. Accuracy should be established across the specific range using a minimum of 3 concentrations across the range, with 3 replicates at each concentration level. Accuracy is usually determined using the data from linearity study. So this can be lumped together, and the data generated in linearity can be used for accuracy. Common mistake in accuracy study is when using a standard that was quantified by the same assay which is being validated. So if you're using a standard in the validation study and the concentration of the standard is measured by the same method which is being validated, then that is one common mistake. So the concentration of the standard should be measured by another orthogonal method. So here are ways to properly determine accuracy. Orthogonal method, as I said, comparison of the results of the proposed analytical procedure with those of a second well-characterized procedure. This is the ICH definition. Example is if the method, let's say if the method being validated is a total protein determination by spectroscopy, then the results of the standard or the results of the validation can be compared with a peptide mapping using LC-MS or a direct amino acid method. An alternative approach is from percent recovery, from spike recovery. Spike a known amount of analyte, which is a standard from a reputable source or in-house or an in-house standard measured by an orthogonal method into the sample or the placebo and calculate the spike recovery. Another way is you start with the high concentration of the sample and prepare a series of diluted test samples, dilute them with the matrix or with the buffer, and calculate the percent recovery. So this applies when there is, if the reference standard is not available. Oh, this is one common mistake that we see in claiming that this statement is ambiguous, which is stated in ICH section 411, I think. Accuracy may be inferred from precision, linearity, and specificity, and it goes further. So without performing accuracy, people say we have performed precision, linearity, and specificity, so we don't need to demonstrate accuracy. So that is a huge common mistake, and that if the method is a quantitative method, then data demonstrating accuracy should be submitted. Also, demonstration of accuracy is required for specificity, or to demonstrate specificity, accuracy should be determined. Another quantity or parameter for validation is range. So range is the interval between the upper and lower concentration levels where the method can be effectively used. According to ICH definition, range is established by confirming that the analytical procedure provides an acceptable degree of linearity, accuracy, and precision. It should be expressed as the reportable results and should have the same units as the specification. If the reportable value in the specification is in concentration unit, mg per mil, then the range should be in mg per mil. If it's in percent composition, then the range should be in percent composition. And again here, range should be evaluated using the data from a representative sample, not from a standard data. So determination of range for some of the method types. For assays, it's 80 to 120% of the target or test concentration. For impurity determination methods, it would be from the reporting level of the impurity to the 120% of the specification. And linearity, accuracy, and precision should be determined over the whole range. So linearity, accuracy, and precision should be demonstrated throughout the range, from the lower level to the highest level in the range. Here are some examples of common mistakes of setting the wrong range. The range was established based on linearity data alone. Instead, range should be demonstrated based on precision, accuracy, and linearity results throughout the working range, including at the quantitation level and the quantitation limit. Another common mistake, assay range reported in terms of response signal, not in terms of the reportable results. So we see that sometimes range was reported in concentration units from 0.2 mg per mil to 10 mg per mil, while the reportable value is in percent composition, like in percent area. So this is a mistake, and the range should be in the same units as the reportable values. For molecular size distribution, the SEC method that we saw earlier in the linearity section, for MSD, for molecular size distribution in purity assays, range should be in percent peak area, not in concentration. The reason I brought it here is because we commonly do this mistake. So since the reportable value is in percent area. And then range should cover the specification, the product specification. So we see sometimes the range validated for the method is narrower than the product specification. That is wrong. So with the minimum, the range should cover the whole specification, the whole product specification. Precision. The ICH definition for precision is the precision of analytical procedure expresses the closeness of agreement, degree of scatter between the series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed conditions. Precision is measured from relative standard deviation, equation that you see there. Repeatability is a form of precision, also known as intra-assay precision, that determines the variation in measurements under the same condition, within the same day, the same operator, the same instrument, and so on, within the same sequence. How is repeatability determined? A minimum of 6 determination at 100% of the test concentration, or a minimum of 9 determinations covering the specified range for the procedure, 3 concentration levels with 3 replicates at each concentration level. My preference is, you perform one big study. It could cover the whole thing, accuracy, linearity, and precision, and so on. So 6 replicates at each of the 6 concentration levels to cover the assay range. which will be a total of 36 determination. Hopefully the assay format allows you to perform that. Intermediate precision, also called as intra-assay precision, evaluates the impact of changing days, operators, equipment, and so on. Poll question number 2, which of the following studies is not expected for method validation? Linearity, specificity, calibration range, and limit of quantitation. If you say C, then you are correct. Detection and quantitation limits. Validation of quantitation detection limit only expected for quantitative impurity methods. The ICH definition for detection limit is the lowest amount of analyte which can be detected but not necessarily quantified. ICH definition for quantitation limit is the lowest amount of analyte which can be quantitatively determined with suitable precision and accuracy. The range and linearity of the method should include quantitation limit as its lower limit. Precision and accuracy should be demonstrated at the quantitation limit. And ICH provides methods for estimation of detection and quantitation limit, but the estimated values must be verified experimentally. Detection and quantitation limits should be in the same unit which the test result is expressed or reported, not in terms of instrument response or other factor. Robustness is mostly studied during method development and optimization. According to the ICH, sometimes it's also called ruggedness, is a measure of method's capacity to remain unaffected by a small but deliberate variations in method parameters, and it provides an indication of method's reliability during normal use. Some examples of robustness study can be, say for HPLC method, minor changes in column temperature, buffer composition, pH, flow rate. For general assays like solution stability over a certain incubation time, extraction time, etc. If measurements are susceptible to slight parameter variations, then the method should be suitably controlled, or a precautionary statement should be included in the procedure. System suitability parameters should be established to ensure minor or unintentional changes during normal usage do not affect the validity of the method whenever it's used. So here is a summary of what we have covered in this brief presentation. What is the purpose of analytical method validation? To demonstrate method suitability through laboratory studies, that the method works as intended. Also, validation is a regulatory requirement. Validation is a proof that the method works appropriately, and only through valid methods can one generate valid data, which ensures safe and effective pharmaceutical products reach the public. Validation studies should be performed via a properly designed study following a written protocol with a preset acceptance criteria. Suitability of the method should be demonstrated using the product itself, not just standard. All validation parameters should be evaluated for quantitative methods. So all the validation parameters should be evaluated for quantitative methods, except for quantitative assay exception is quantitative assay methods don't need detection limit and quantitation limit determination. References. Contents of this presentation was based on the references listed on this slide here. Hence, this guidance and guideline documents ought to be the starting point for any validation exercise. This is the end of my presentation, and thanks for your attention.
Okay, so I'd like to thank our speakers and start the question and answer session now. So the first question goes to Alyssa, and that's how can a manufacturer or sponsor determine which FDA center their medical device should be submitted to?
Great, thank you for that question. One thing that sponsors can do is to search the product classification database at fda.gov and check the types of device the type of device you are developing that could already fall under an existing device product code. The description for each of those product codes will include an indication of which center reviews the devices under that product code. But if it is unclear to you which center has jurisdiction over your product, you could consider contacting the Office of Combination Products at FDA, where you can request a formal or informal feedback on the classification of your product, either as they'll determine if it's a drug or biologic or device or combination product, and they'll also determine which center at FDA, so CDER, CBER, or CDRH would regulate that, and that's through a request for a designation process, also termed RFD. Thanks.
Great, thank you. The next question goes to Mikhail, and this is a question that's actually close to my heart as well. So could you give us your expert opinion about the safety risk considerations on the use of UK plasma for manufacturing plasma-derived medical products, specifically related to concerns about transmissible spongiform encephalopathies?
Thank you very much for this question. I need to start with a disclaimer. This is a regulatory question, and I will not be able. Please don't use my answer as regulatory guidance. If you so much as think of using non-US plasma, that is plasma obtained from donors who are not from the United States, if you are so much as thinking about that, please speak with the Food and Drug Administration about your assessment of risks first, before you go into an expensive risk product development exercise. Now, from my opinion as an expert, I'd say that it would be really hard to justify the use of plasma of donors outside of the US and specifically from the UK because of the risk of prion contamination. Theoretically, one may consider a very unique product that meets a very unique clinical need that is unmet, and a manufacturing process that is specifically developed to reduce the risk of prion transmission. We also may think about the evidence that is obtained in recent years about the risks associated with prion occurrence. But I think that this is a very high bar to meet, and I have no experience with products like that. I also know that all manufacturing facilities that happen to use plasma pools of donors from the US and plasma pools from other jurisdictions, they happen to have very strict procedures, changeover procedures that would prepare the equipment and the facility to the use of plasma from non-US donors and switching it back to US donors. In fact, in most cases, it becomes easier to dedicate equipment to the products that are made with the US plasma. Thank you.
Okay, thank you for that answer. So the next question goes to Emnet. What's the difference between method validation and verification?
Thank you for that question. So something that I forgot to mention early on, like Mikhail, is the disclosure portion in my presentation that I did not put up. By the way, my the opinion in this presentation that I just did is on my own, and it's not reflective of FDA. So I'd like to highlight that. Going to that question, so the difference between verification and validation is verification is usually performed for compendial methods, which are already in USB standard methods. As I mentioned in my presentation, for verification, you probably have to do repeatability and write up your SOP according to the product and the instruments that you use. But for validation, usually is methods that are developed in-house. So depending on the method type, you have to go through validating the whole validation characteristics, specificity, accuracy, and so on and so forth, depending on the method type. Thank you.
Okay, thank you. The next question goes to Alyssa again. So if a product delivers a biological product on a scaffold, would the combination product be submitted submitted under a BLA to CBER, or would it be submitted in a device application to CDRH with both components listed in the applicable application?
Great, thank you for that question. That's a great question. So for combination products, typically the decision as to which center, either CBER or CDRH in this case, would regulate the product does rely on determining the primary mode of action, or the PMOA, of the product. So combination products are defined within the CFR, 21 CFR 3.2E, and that's typically a good place to start to determine what center would regulate that. There's also a lot of helpful resources about combination products on the FDA public website. But again, like I mentioned before, you can also contact the Office of Combination Products for an RFD to get a formal or informal feedback on your product, if it's going to be a drug, a biologic, device, or combination product. And then again, they'll render a decision on which center the FDA would regulate it. So I guess in this hypothetical case, if the biologic is the entity that imparts the therapeutic action, and then that's being put on a scaffold and then administered to the patient, then most likely this combination product would be regulated by CBER. And thus the product applications, if it's an investigational stage, that would be an IND, or for commercialization, that would be a BLA. Those would be submitted to CBER. And the scaffold information that's part of the combination product should be included in those applications, and that would be reviewed in its entirety within CBER. Thanks.
Okay, great, thank you. The next question goes to Mikhail, and that is, how do we justify contaminant limits in a product?
Thank you. So, well, a simple answer is that contamination of the product, it makes this product non-compliant, and this product should not be allowed for distribution. So that's the difference between impurities and contaminants. If we are specifically talking about contaminants, then there are no contaminants in the product. There are no specific limits on the product that allow for contamination. Now, we can talk about the limits that are derived from the assays, suitability of the assays. All assays can give us an answer only to a certain limit, and those limits may be developed. But in principle, contamination is something that is not allowed in the product. For example, mold. We understand that mold exists everywhere, spores and everything. But your facility should not be a source of mold. At least it shouldn't be a source of mold that exists in such quantities that mold can get into your product. As such, a specification on mold may be interpreted as your inability to maintain your facility without mold contamination, which would indicate that your procedures for cleaning and validation of the procedures that you use to remove mold spores from your facility and your equipment are inefficient. That would be something that you want to avoid. So instead of implementation, in this example, of a specification for mold, you should spend your efforts on demonstrating that mold is absent from your facility and your facility does not support mold growth and mold does not get into your product. So the short answer, you don't set specifications for contaminants because contaminants shouldn't be there in the first place. You should spend your efforts on preventing contamination of the product. Thank you.
Okay, thank you. The next question is a good one for Eminent. So can you give an example of what data you would want to see to support linearity of a method like cell count of a number of cells in a suspension? And how would that be different from accuracy data?
So this is so cell count. So I'm a medical chemist, right? So, but there are parallels in here, like for example, for particle size methods and things like that. So, there is no linearity per se to evaluate on those type of methods. So, I think it would be similar for cell count methods. There's nothing increased proportionally. There's no linearity like a concentration measurement would be. So I would there has to be a USP standard method or a 1000 method, I would say, which may deal with this. I'm not familiar with cell count assays, but there is a correlation or similarity to particle size methods that measure size, and I think you should consult to a USP method. There should be something in that regard, I think. But good question. Thank you.
Okay, thank you. So the next question is for Alyssa, which is for device biologic combination products, where the biologic is regulated by OTAT, would the device constituents always be reviewed by CBER, or would they sometimes be reviewed by CDRH?
Thank you for that question. Yes, typically, if the application is coming to CBER, the device constituents are reviewed as part of that biologic submission. So may it be in the clinical trial application, which is the IND, or in a biologics licensing application, which is the BLA. And depending on the device constituents, CBER can review those, but we can also consult our device colleagues in CDRH internally, especially if the device constituent is novel and probably hasn't been approved or cleared. Thanks.
Thank you. So for Mikhail, where can we find information on allowable safe limits for impurities in CBER products?
That's a good one. Thank you. It really depends on the product. And in general, it is the responsibility of the product manufacturer to demonstrate that the product which is manufactured with the manufacturing process that is developed for this product is safe and effective. Depending on the indication for the product, we may consider different allowable tolerances for impurities, and that very much depends on the type of the impurity. And because we are talking about the large number of products and impurities, it is impossible to show a single place for you to look for this information. This is the responsibility of the manufacturer to understand their product, to understand the critical quality attributes, and develop the levels of the specifications for the levels of impurities that support the indications being sought. You should discuss any concerns that you have with the Food and Drug Administration. You probably know that there are different levels of engagement. Some simple questions may be answered relatively quickly. Other questions need to be asked as part of a meeting package. I leave this to your regulatory team to decide on which pathway for the communication with the agency you will choose. Thank you.
Thank you. And now a question for EMNET, which is literally the question I get asked at every single meeting I attend, which is, could you elaborate on the difference between validation and qualification?
Yeah, definitely. I would guess people would ask that. So as a regulatory, I'm speaking disclosure. I'm speaking my opinion, not of FDA. So in any of the regulatory documents and guidelines, if you see, there's nothing about method qualification. So when you bring your finished product in BLA or whatnot, the method has to be in a state of completion, in a state of being validated. So if you reach that stage, then you have to fulfill the requirements which are stated in the guidelines, in ICH guidelines. So there is no pre-validation or qualification, or the semantics is not covered in guidances and regulatory documents. So it is probably according to whatever you think is appropriate for your product, I guess. Sorry, I probably did not answer it properly, but that is what it is. Thank you.
Okay, and I think that will be the last question for today. So I would like to close out this session and this question and answer session by thanking the audience for their questions and by thanking our speakers again for their presentations and clear answers. Now I'm going to hand off to Dr. Larissa Leptiva from the Office of Tissues and Advanced Therapies for her remarks to conclude this conference. Thank you.
Thank you, Graham, and thanks to all of our session participants. It's been very enlightening to hear some of the perspectives shared in relation to product quality assurance and a number of other topics that we have discussed today. We have now come to the conclusion of the CBER track and, in fact, to the conclusion of the entire week-long conference, which included presentations and panel discussions from the Center for Drug Evaluation and Research, Center for Devices and Radiological Health, and Center for Biologics Evaluation and Research. As in previous years, we hope that the information and the resources which we've shared, as well as the presentations and our experts' recommendations and responses to questions, were helpful to you, and that you will make use of the resources provided by each center and by the Small Business and Industry Assistance Program when you are developing new products, new drugs, biologics, devices, tests, or any new technologies that will help make people healthier and safer. I would like to thank all of our presenters and moderators for sharing their expertise and valuable perspectives, as well as each center's planning committee. We had a number of those, and the SBIA program staff who worked long hours to organize this event and made sure that it ran as smoothly as possible in the virtual environment. And very special thanks should go out to the conference support team for all of the preparation and the excellent job that they've done during the conference, particularly when handling all of the expected as well as some unexpected events and handling them with poise, humor, and confidence. This year is the second time when we were again able to have 3 medical product centers participate in the annual REdI event. And on behalf of the Center for Biologics, I would like to express our sincere gratitude to our sister centers, Center for Devices and Center for Drugs, for their support and for this very productive collaboration, as well as to the SBIA staff for their experienced organization of this event. They do a great job not only with this conference, but with many other conferences oriented to small business audiences. But most importantly, I would like to thank all of you who have attended and participated in this conference. It is your participation, your engagement, and your unwavering interest and questions that make this event successful and drive the contents of this conference every year. And we hope to continue meeting your needs for the regulatory educational experience so you can translate it into efficiency of your product development. If you missed any session or presentation in this week's event and you would like to view it, or if you would like to listen to some of the talks again, you can access our website shown here on the slide to catch up with the contents of the presentations. Please also visit CBER's OTAT Learn website, which I'm sure you've seen on multiple slides yesterday and today, for more educational materials on regenerative medicine therapies and other CBER and OTAT regulated products. And of course, the Regulatory Education for Industry Conference is an annual event in which, as we saw on Monday in the polling results, many of you participate regularly. So we invite you to join us again next year for more educational series on new topics and issues essential to the development of drugs, devices, and biologics. We look forward to seeing your evaluations and recommendations for new themes to include in our next year's conference. Once again, thanks to everyone for joining us this week. We wish you all a restful weekend and a productive year ahead. Thank you.